Hi everyone, welcome to the educational course on time-varying connectivity in resting state fMRI. So this is actually the first presentation in this course, and the aim of this presentation is to give you a broad and comprehensive overview of the theoretical and methodological frameworks that are being used in literature in order to assess the time-varying behavior of functional signals in the brain. So in this talk, we will first discuss the basic concepts of functional connectivity, and then we will define the proper terminologies that differentiates these methods, and also in order to avoid confusion in the succeeding talks of this course. So first off, functional connectivity is defined as the statistical dependence between functional signals stemming from different brain locations. And how we compute that is that we divide the brain into different regions, and then we look at the intensity of signals in each of these regions. And then we summarize the interdependence between these regions using an average measure known as the correlation or the Pearson correlation. And if we do that over all regions of the brain, we obtain a pairwise set of correlations known as the static functional connectivity in the form of a connectivity matrix as shown here. So there are two different properties of static functional connectivity that I would like to address in this talk. The first is that basically static functional connectivity gives an average measure over the whole acquisition length. What does it mean? So given two time courses, the output of this is a single measure. And so even in the context of resting state fMRI, even though we obtain different frames, brain scans at around 2 seconds, and we do that for the whole 5 to 15 minutes, the whole output of static functional connectivity measure applied to this data is a set of correlations in the form of a connectivity matrix. Meanwhile, another important property of static functional connectivity is that it is memoryless, which means that if we are given 2 time courses and we shuffle the signals across time, the value of the connectivity that we get, the correlation that we get, is still the same. This is because a correlation by itself is invariant to the reordering of the data that we are given. So here is an example of a minimally preprocessed fold signal. And as you can see, just looking at it by the naked eye, you can already observe several fluctuations at different regions of the brain. And sometimes there are special brief instances where these regions of the brain are co-activating or co-deactivating. And so if we look at it, and if we know that only an average measure is obtained by static functional connectivity, there's a lot of information that is missing when we apply a static functional connectivity. And so in order to capture the rich information in the brain, brain function, we need to go beyond static functional connectivity and consider advanced approaches that look at the fluctuations of the signals. And detaching from the average measure of static functional connectivity, time-varying functional connectivity approaches such as sliding window analysis and co-activation pattern analysis addresses this concern. Meanwhile, detaching from the memoryless property of static functional connectivity are dynamic functional connectivity approaches such as autoregressive models and ICAPs, or innovation-driven co-activation patterns, as well as spectral approaches. Therefore, in this talk, we will discuss these methods, in particular the time-varying functional connectivity approaches and the dynamic functional connectivity approaches. And then lastly, we will touch upon the discussion on how the underlying anatomical structure affects or constrains the dynamics of brain function in the using different methods that are currently very prominent in the field. Okay, so to start, we first discuss the most popular method that is used in under time-varying functional connectivity. So similarly with static functional connectivity, we start with the intensity values or the temporal signals of the bold data across different regions of the brain. But instead of looking at the whole duration of the recording, we look at individual time windows that is then scanned through the whole duration of the scan. And the output of that is a sliding window correlation signals that are varying in time. And so if we do that across all the whole brain, we obtain a set of functional connectivity matrix at each time window. So given this framework, we can then analyze further the temporal fluctuations of these interdependence between regions of the brain. And to do that, we usually perform the so-called connection-wise variability, which is basically computed by taking the standard deviation of the, for instance, of the sliding window correlation across time. And another method is to look at the temporal, the functional connectivity matrix as a set of graphs. And so what we can actually do is to evaluate the evolution of the graph metrics that is applied at each functional connectivity at each time window. So for instance, we can then obtain a set of time varying modularity measures of the graph being considered. And another way, another subsequent method that can be applied is that we can decompose this functional connectivity matrix to obtain connectivity states. And the interpretation of these connectivity states depend on the technique that we use to decompose these functional connectivity matrices. So for instance, Allen et al. In 2012, decomposed these functional connectivity matrices into connectivity states using K-means clustering, while Leonardi et al. Decomposed these connectivity states using principal component analysis. Furthermore, these connectivity states can then be further analyzed by looking at their temporal properties. So depending on the technique that we use to assess the temporal properties of this signal, of these connectivity states, for instance, using K-means clustering, you can actually then obtain a set of states that are exclusive in time, meaning only one state is happening at a single time point. And so with these, we can compute their transition probabilities. Meanwhile, if we are able to obtain states that can jointly occur at a single time point, then we call them the meta states, which means that at a single time point, the behavior is described by a combination of multidimensional connectivity states, in this case known as the meta states. And then, of course, from that, we can compute their occurrences and then co-occurrences. So far, I've told you that sliding window analysis is the most popular method that is being used in time-varying functional connectivity studies. However, even though it's the most popular, it suffers from severe caveats and limitations. And the most common one and the most apparent one is the choice of the window parameters. So of course, depending on the length that we use, there's a lot of information that we can either get or we can miss. So therefore, what we have to do is to obtain the optimal window length that gives a proper trade-off between sensitivity and specificity, which means that we need to have a window that is short enough not to miss the real functional connectivity fluctuations. So this, of course, can depend on whether we have an idea of the timing of the events that we are interested to study. And on the other hand, we also want to have a window that is long enough to detect only the real fluctuations in the bold data. So it's already been found that there's been a spurious fluctuations that is included in the data depending on the window that we use. So it has been shown by Leonardi et al. That in fact, the minimum window that has to be used when performing sliding window analysis is 1 over the minimum frequency that is present in the data. And this is, of course, dictated by the pre-processing steps that we applied on the data itself. And then, of course, another important aspect of the parameters that are being that has to be overcome is the window shape. So the most commonly used shape for sliding window analysis so far is the rectangular one. But other studies have also tried to explore using tapered windows, which give less importance on the boundaries of the windows. And perhaps a very important one is the choice of the parcellation. So this limitation of sliding window is not only limited to this method itself, but also in general to the whole neuroimaging. This is because parcellation can be obtained using different methods. It can be functional based or anatomical based. And so in order to solve this limitation, what has been recently done in Dimitri Vanderbilt's lab was to, instead of using region-wise parcellation, what they did was to basically look at the time-varying behavior of functional connectivity at the voxel level, which means that instead of using regions, we look at the individual time courses at each voxel. So then you can imagine that we have a huge matrix of voxel by voxel at each time window. And therefore, this poses a problem of computational complexity and also the storage itself. And so what they did was to summarize the dominant pattern in these individual connectivity patterns at each time window using the eigenvector centrality. So the eigenvector centrality is just the eigenvector that gives the most variance or the most dominant pattern that is present in the data at each time window. So from the eigenvector centrality, they were able to parcellate the healthy brain into different regions. So as you can imagine, time-varying connectivity information was not necessary at the voxel level because the neighboring voxels are more likely to be correlated. And as such, in this work in the lab, they merged together voxels that show similar pattern, and they used the information to derive 37 long-range parcellation in the brain, which can then be further subdivided into 449 contiguous regions. So this makes the method completely data-driven and also solves the well-known issue of the choice of the a priori atlas. And of course, very importantly, it includes already the information about the dynamics of functional networks themselves. Okay, now going beyond the study of sliding window analysis are the methods that evaluate temporal time-varying functional connectivity at the frame-wise level. So what do I mean by this? So recall that static functional connectivity look at the average, gives a single measure across the whole time course. And when we do sliding window, we improve the resolution by zooming in into a specific window. However, this also suffers from low signal to noise ratio and potential aliasing depending on the frequency present in the data and the window that we use. So what frame-wise analysis does is to zoom in at the maximum resolution of the fMRI data, which is the frame-wise resolution, and considers only key points in the fMRI signals that are considered important. And so one of the most prominent method that does this is the co-activation pattern analysis, which looks at the maximum, looks at the significant time points that have significant activity. So this is inspired by the point process analysis that is introduced to us by Taglizitti and colleagues. And in order to obtain these co-activation patterns, what Liu and Duan did in 2013 was to initialize a seed gold time course, given by, in this context, the posterior cingulate cortex. And then they look at the significant time points according to the posterior cingulate cortex. And those time points that are above the threshold are fed into a classification algorithm known as the K-means clustering in order to obtain the different co-activation patterns in the data. So in this case, here are the co-activation patterns according to the posterior cingulate cortex. And even though we use the posterior cingulate cortex, we don't only see the default mode network itself, but also different subsystems of the default mode network at different temporal frequency. And another method that is under frame-wise analysis is the leading eigenvector dynamics analysis that was introduced by Joan and Cabral, who is also going to present in this course. So what they did here is that they got rid of the window by computing the connectivity using an instantaneous coherence connectivity method. So this method leverages on identifying the phase coherence between signals at each region and then at each time point using the Hilberton's form of the fMRI time courses. So doing it at pairwise regions yields a coherence connectivity matrix whose temporal evolution can then be assessed. So as you can imagine, similar to the voxel-wise sliding window functional connectivity data, this method also suffers from a dimensionality problem. And as such, each brain connectivity of each window, of each time point, I should say, is approximated by the leading eigenvector in order to obtain the representative dominant pattern at each time point. Okay, so going over now and recalling that there are two important properties of static functional connectivity. One being average, which is taken care of by time varying functional connectivity approaches. There's also the property of being memoryless. And in this case, dynamic functional connectivity approaches takes this into account and overcomes this problem by making sure that the influence of subsequent time points are taken into consideration. So recalling in CAPS, we obtain frame-wise analysis. So the same is true, is being done in innovation-driven co-activation patterns, but instead of using the peaks of the time points, peak data or the significant data, significant frames, we instead use the innovation. But then what do we mean by innovation? So innovation is obtained here by first deconvolving and denoising the bold signal in order to obtain a black type activation. And then these signals that are already denoised and deconvolved are taken the derivative of. So we take the derivative of the signals to obtain the time points that detects the transient brain activity. So these are moments of activity changes in the bold signal. And so as you can see here, these black time points, these are known as the innovation signals. And the time points where the innovation signals are significant then undergoes a temporal clustering procedure using, again, K-means clustering to obtain the different functional networks, as can be seen here. And these ICAPs are further assessed and are a further process in order to obtain the activity time courses for each of these networks. So what we actually did here to apply this method was to use in a recent paper that we published, is that we applied this method in a data of 45 hours of sleep and wakefulness recordings. So as you can see here, even if a person is sleeping, the brain is still very active. So we can see a lot of activity that is happening. We can see the different well-known resting state networks that are activating and co-deactivating, co-activating at the same time. And given this huge amount of information, we can then actually evaluate how many times do each of these networks co-occur and how many times do each of networks occur at different sleep stages. And so this is actually what we did here, was that we evaluated the amount of overlap between these networks. So each line that we see here is a set of ICAPs, and we count the number of ICAPs that are overlapping with each of the ICAPs that we consider. And this is done across wakefulness, stage 1, stage 2, and stage 3 of the non-rapid eye movement sleep. And as you can see, surprisingly, what we observe is that the most number of overlap actually occurs in stage 2, which is a bit surprising given the fact that we also observe that the functional connectivity between these networks actually decreased from wakefulness to stage 3. So, and also that the normalized mutual information between them is actually decreasing. So what we observed then in this recent work of ours is that there seems to be a paradox, that the brain is very unstable during stage 2 and very stable in stage 3. And that because even though there is a high attempt for these networks to communicate during stage 2, the efficiency of information transfer is still small. And this is reflected by the functional connectivity and the normalized mutual information. So this is a very important finding that would have otherwise been missed if we use static functional connectivity, which just looks at the average value or average behavior of the signal at each stages of sleep. Now, regarding temporal modeling, one of the most prominent methods that overcome the memoryless limitation of static functional connectivity are autoregressive models. So in autoregressive models, we model the fMRI volume at time t as a linear combination of signals from p previous time points. So the p here is the model order that we use. And here, the matrix A is a matrix of coefficients that is given by the dependencies between signals from time t and previous time points. And of course, E here is the independent zero mean Gaussian noise. And what they have found actually in the works by Raphael Liegeois, who is also going to present after this talk, is that first order autoregressive models explain functional connectivity fluctuations better than static functional connectivity and hidden Markov models. And it's actually even more interesting is that they also found that static connectivity and dynamic functional connectivity metrics could actually give complementary information in the sense that task-based phenotypes that are characterized at faster timescales are better captured by dynamic functional connectivity, while other types of behavioral phenotypes, such as self-reported measures like loneliness or satisfaction, are equally captured by static and dynamic functional connectivity approaches. So overall, they found that phenotypic measures, dynamic functional connectivity actually do explain more behavioral variance than static functional connectivity. And autoregressive models have also been employed to retrieve the dynamic extension of component analysis known as the dynamic mode decomposition. So in this case, it is the matrix A that is decomposed to obtain the spatial information using an eigendecomposition of the A, where the spatial information is given to us by the eigenvectors of A, and the temporal information is given to us by the eigenvalues of A. So in this case, the spatiotemporal modes that are obtained are characterized by the damping parameter and as well as the associated oscillation frequency or period t. So for example, we show here some of the dynamic modes that were obtained in their paper. So very prominently, we observe the default mode network as well as its counterpart, which is the task-positive network. And in dynamic mode 2, we obtain subsystems of the default mode network as well as the visual system. So similarly, with co-activation pattern analysis and innovation-driven taps, we also have access to subsystems of the default mode network, which would have otherwise been missed if we use static functional connectivity approach, which just takes the average connectivity. And lastly, now we touch upon the effect or the influence of structure in the time varying behavior of brain function. So two of the most commonly used now recently in the field are graph signal processing and whole brain computational models. So I first start with graph signal processing, which is very emerging in the field that is borrowed from the engineering from engineering. So basically what they do here is that we define a function atop the node of a static structural backbone. So what we do here is we define the graph from the structural connectivity that is obtained from the fusion MRI data. And then on top of this brain graph, we actually define our graph signals coming from functional MRI atop the nodes of our brain graph. So basically, we try to manipulate the signals coming from the functional MRI on top the static backbone, which is given to us by the structural connectivity from the fusion MRI. And from the structural connectivity, we can then assess the behavior of functional data by first decomposing the structural connectivity into its constituent eigenvectors. So this is done by decomposing structural connectivity, the Laplacian of the structural connectivity, and looking at the eigenmodes, which was first introduced to us by Atasoy and colleagues. So these eigenmodes, or the structural harmonics, describe to us the spatial patterns that are present, that are inherent in the structure. And as you can see in this presentation, you could see that for the first eigenmode, we observed a constant spatial variability. As we increase then the eigenvalues that we consider, there's a lot more variability that is being observed in higher order eigenvectors or eigenmodes. And in the recent work in Dimitri Van Poel's lab was that they actually expressed functional signals that are defined on the nodes of the graph as a linear combination of the structural harmonics. And then they filtered this data according to two different descriptions. So one are the first one are the low frequency components, and the other are the high frequency components. So what basically they did here was to only consider low frequency considered components, being the ones that are most coupled with structure or more aligned with structure, versus the high frequency components that are less aligned with the underlying structure. And then they introduced a single metric known as the structural decoupling index that takes the ratio between the decoupled versus the coupled. And what this single metric does is to give us an information which regions in the brain are most likely decoupled, which functional signals defined on each region of the brain are most likely to be coupled or decoupled with structure. And what we found here is that, very interestingly, so the blue colors here given by are the coupled regions. What we find is that sensory regions such as the occipital or the pre and post central, as well as the temporal lobes, the temporal regions, are colored blue, which means that these regions are more likely to be coupled with the underlying structure. Meanwhile, high-level cognitive regions, such as those that are associated with multisensory integration, memory, and decision-making, are actually decoupled with the underlying structure. And if we feed this information into a neurosync meta-analysis, where we look at the percentile of the structure decoupling index and plot that with respect to the topical terms that describe to us the different cognitive processes, we observe that indeed low decoupling index are associated to less complex tasks, whereas higher order cognitive tasks are more associated with more decoupled index. So indeed, we here find that the influence of structure onto brain dynamics is actually behaviorally relevant. And so having been able to extend analysis from region-wise analysis to voxel level, we also tried to introduce a new avenue for graph signal processing by not only doing it at the region level, but also at the voxel-wise level. So here in a recent work, we actually introduced a brain graph that is defined at the voxel level, meaning that all voxels in the brain, including the white matter, is included. And if we decompose this voxel-wise brain graph into its associated eigenvectors and eigenvalues, what we find here is a set of human brain eigenmodes at the voxel level. And what we find is that the first few eigenvectors are describing the head shape. So you can see there's the posterior and anterior and the left and the right. But as you, of course, increase the eigenvalue, the associated eigenvectors describe more spatially varying spatial patterns. And using this voxel-wise brain graph, we were able to interpolate signals in the white matter. So in this case, what we did was to mask gray matter Bolt signals, and then we define our gray matter signals onto the nodes of the voxel-wise brain graph, where we keep the values of the gray matter and we keep all the other values of the white matter to 0. And when we did that, we were able to interpolate the Bolt signals across also towards the white matter. And the information that is given to it, that it gives us, is the following. So if given that we have a high signal on one region of the brain, in this case, the posterior cingulate cortex, and another region of the brain, which is the medial prefrontal cortex, what we find is that we actually observe white matter structures that are mediating the interaction between these coherent brain regions. And if we do that using PCC-SID connectivity, we also obtain not just the default mode network, but also the patterns that are white matter structures that are associated in the recovery of this network. And similarly with CAPS, where we were able to obtain, as you can recall, the different subsystems of the default mode network, we also did that through the in-painted signals. And here we actually observed not just the coactivation patterns in the gray matter, but also the coactivation patterns that includes the white matter, which then gives us an idea which of the white matter structures actually give rise to this dynamic repertoire of functional data. And then lastly, there's also the family of methods that addresses structure-function relationship, which are called the whole brain computational models. So this family of methods simulate functional signals starting from the anatomical constraints given by the structural connectome, and also, of course, given by the models of neural dynamics. So several methods have already been introduced before, and all of these works all point out to specific characteristics of the model, which realistically simulate functional data. So basically, these are the requirements that the simulated data should satisfy. So first, that the functional connectivity spatial patterns should not only be reproduced, but also that the whole frequency spectrum of the resting state signals should also be realistic. More importantly, in order to understand the underlying mechanism of functional brain dynamics, it is then important for these models to also capture realistic temporal evolution of the data. And with this, I summarize my presentation by the following 3 points. We have found that the brain is always active and actually very dynamic, that spontaneous brain activity is present even during sleep. And what we find is that they do change over time. And in fact, we actually observe that the brain is even more active during sleep. And another point is that there's a plethora of methods that can be used depending on the research question that you want. And so we adapt the method that we use depending on the questions that you want to answer. And we also have different approaches that could be either data-driven or model-based, or dynamic or time varying. And it's also important to note that until now, windowless methods are still under use in clinical applications. And so it would be nice to further these developments in order to apply these methods in the clinical settings of clinical population. And one last important aspect is the integration of structures. So we have found behavioral relevance of looking at how the underlying anatomical bedrock influence the recovery of dysfunctional networks, of the dynamics of dysfunctional networks. And so it would be a very good avenue for continuous research in the field of fMRI. With that, I thank you for listening.
Hello, everyone, and welcome to this second talk of the time-varying functional connectivity course. Welcome as well to EPFL with a nice view on the campus and the Geneva Lake behind me. So during the first course, Anjali presented the main and the most important concepts to, for example, distinguish between dynamic and time-varying functional connectivity, presented also the main methods that have been developed to explore dynamic properties of fMRI time series, but also functional connectivity time series. And in this second presentation, what I want to do is to have a focus on how to better characterize the nature of these dynamics that we observe in fMRI time series. And to this end, null models have been used quite a lot, especially quite recently. For example, I'm sure you've heard of phase randomization. But I think the interpretation of these models and the results is not that obvious, not that straightforward, and should be done with caution. So that's why I've designed this talk. And let me just say that it's essentially providing the main messages of this paper that we wrote with Thomas Yeo and Dimitri van der Wiel, which has been submitted as a preprint recently and submitted for publication. So maybe by the time you hear this, it has been published, hopefully. Okay, so this is the outline of the course. I will start with just the basics of null model testing so that everyone is more or less on the same page with the same methodological tools to understand what we are doing when we do null model testing. Then I will present a few null models for fMRI time series. I've mentioned briefly phase randomization, but there are other models that could be used for exploring properties of fMRI time series. And then I will discuss how we interpret the results of null model testing in terms of functional dynamics. And you will see it's not that easy, and it should be done with caution. So let's start with the basics of null model testing. As a researcher, what is the type of question we might have when we have to treat, when we have to analyze a time course? Well, one of the questions we have is, what is the statistical nature of this data? And to answer that question, actually null model testing could be interesting. So how does null model testing work? If we call this the original data, which is not a very revolutionary nomenclature, what we want to test when we do null model testing is whether a model M is good to model to represent this original data. So what we do in surrogate testing or null model testing, that these are equivalent words, in at least I will use them in the same context. What we do is that we identify a model from the original data, the one that we want to test whether it's sufficient to represent the original data. And from this model, we generate a lot of data with, for example, random input noise in the model. And then we compare what we call surrogate data to the original data. So how do we compare these two data sets? Well, we essentially define metrics that are to be computed in the original data and in the surrogate data, and we discuss the comparison between the original metric and the metric in surrogate data. So let me precise here that the metric in original data is usually only one metric, so one value. If it's a scalar, it would be one value of that scalar. Whereas in surrogate data, since we have the possibility to generate a lot of surrogate data, we have a distribution of surrogates of this metric in surrogate data. So if M O, the value of M in the original dataset, is found to be different than the distribution of values that we have in surrogate data, then it means that the model M, big M, is insufficient to fully describe the original data. On the contrary, if MO is found to be not different or to be equal, but to be not different from the distribution of values that we have in surrogate, then it means that there is no evidence that the model capital M is insufficient to describe the original data. Importantly, and maybe it's a bit frustrating, but this does not mean that the model M fully describes the original data, right? It just means that there is no evidence that M is insufficient. And from what I know, yeah, there is no real way to definitely show that a model completely describes original data, right? Okay, let's consider a first example to put that into practice and to maybe provide you with more intuition on how null model testing works. Let's consider that we want to test whether the model M, capital M, being the univariate Gaussian model, is good to describe the original time series. So these are the original time series, right? The model M is that each time point has been generated by a Gaussian distribution with a mean mu and a standard deviation sigma. So here it's important to understand that the horizontal axis is time, and this model assumes that each time point at each time step, it's generated separately as a realization of a Gaussian distribution. So in particular, there is no link between successive time points. So in the surrogate data, each time point has been generated separately. So in the original data, there might be connection between successive time points, but here there is no autocorrelation in the data. Okay, so you will see later why I have kept a line for operation on data, but the important point here is to understand what original data properties have been preserved under this null. Here it's quite simple. It's mu and sigma, the mean and the standard deviation. And a test measure M, so the variable M presented in the previous slide. So an example of value of useful value M could be, for example, to compare the counts of threshold crossings in original and surrogate data. Could be also to compare kurtosis. So kurtosis is defined is a fourth order statistics of the data. And you could define many other statistics and useful values of M, but obviously nothing that involves mu or sigma, because these are by construction the same in original and surrogate data. And so you would have that the null model is not rejected, but for obvious reasons, right? It's not going to be very informative about your data. So conclusion for this, what is the conclusion that you can draw from the test outcome? Well, if you find that MO is different from MS, MS being again a distribution of values, it means that data cannot be fully explained by a Gaussian, or in other terms, that mu and sigma are not sufficient to fully characterize the data. If, on the contrary, MO is equal to MS, then there is no evidence that data cannot be explained by a univariate Gaussian distribution. So I hope this example is quite clear. It's the simplest example we'll have in the course. And let's now move to another case, which is related, but not doing exactly the same thing. So let's consider the case where we generate data, surrogate data, by shuffling all the time points. So that means that we take the original data and we just shuffle all the time points to generate different realization of surrogate data. In this case, I want to ask you, what is the null model M associated to this operation? So here the null model is implicit, and that's why I kept a line for operation on data. But it's important to be able to identify this null model, because as you will see later on, this will allow to clarify what is the statistical information of the original data that we are actually trying to characterize. Okay, so what is the null model M associated to this operation? Is it A, the same as in the previous example, but just using another procedure? Is it B, that data is IID, so meaning that IID is independent and identically distributed, meaning that each time point has the same distribution, but is independent from each other, and with a distribution matched to the original one? Or is it C, that data follows a first order autoregressive model? Okay, take a few seconds to think about it. Okay, so let's go step by step. If we define and if we show here the distribution, the sample distribution of the original data. So there are a lot of values around 0 and less values at the extreme values. Do you think that this distribution is affected by data shuffling? I think it should be quite clear that it's not, right? So that means that each of these surrogate datasets will have exactly the same distribution as this one. And hence it means that the correct answer is B, data is IID with a distribution matched to the original one. And we'll see that in particular, the mean and the standard deviation of the original data is preserved, but also all other statistics defined by this distribution. Okay, so it's not A, the correct answer. And coming back to formally defining the null model, if we say that this D is the distribution, the null model would be that XT follows that distribution for any time T. The data being preserved, as I said, is mu sigma, but essentially any statistics computed from D. So in particular, mu and sigma, but also kurtosis and any other exotic metric you could imagine from and build from D. Examples of useful values of M would be, for example, again, to compare the counts of threshold crossings between original and surrogate data, which somehow is a proxy for autocorrelation structure in the data. You could directly compare the autocorrelation structure, etc. But again, nothing involving D, because otherwise this would be by construction the same in original and surrogate data. How do you interpret the outcome of a test? Well, if the values in original and surrogates are found to be different, then it means that data contains some form of autocorrelation. And on the contrary, there is no evidence that temporal ordering is important if you find no difference between MO and MS. So this is it for the first toy examples. But I want here to make a few remarks. So first, null hypothesis testing is different from relevance testing. And I will get back to this a bit later, but maybe to provide you with some intuition of what I mean here is that in some papers, I read that null model testing has been used to check whether brain dynamics are significant in this region or that region or things like that. That's not what null model testing is doing. Null model testing is just a tool to better characterize the nature of the dynamics that we observe. A dynamics could be linear, could be Gaussian, could be nonlinear, non-Gaussian. But so rejecting the test just tells you whether it's, for example, nonlinear, non-Gaussian, and things like that. But it's in no way a test that should be used to assess relevance of a given metric, for example, to assess neuroscientific relevance of the metric. But we'll get back to this later. Then just a brief word about stationarity that is also being used sometimes a lot and sometimes not with the proper statistical definition that it should come with. So stationarity is an assumption that comes with models. It's not a property of data, it's a property of models. And for example, in the two first cases, the two toy examples, these two null models are stationary. And so rejecting the null, you could interpret it as a non-stationarity, but there are many other causes for rejecting the null models. For example, the data not being Gaussian, the data not being linear, etc. And likewise, there are many different models and complex models that are stationary and still exhibiting very sharp transition between different states. For example, hidden Markov models are stationary, but yet showing this sharp transition between states, for example. And finally, and maybe most importantly, the fact that rejecting a null model does not mean that the model is useless. And just to give you some intuition on this, consider the following example of the distribution of women hate in the US. So horizontal axis is the hate and vertical axis is the percentage of population. Let's now test whether a Gaussian model is good for this to represent this distribution. So let's test whether the null model M is able to describe the distribution. So to do this, that's the first example that we took. We have to identify a Gaussian model from the data, which gives us the following model represented in blue-green, with a mean of 64 inches and a standard deviation of 2.7 inches. So that's the average and standard deviation of women hate in the US population. Let's now consider the following statistics, which is that the count of individuals, of women taller than 75 inches. Under the null, and if you do the math, it's straightforward to observe it, but under the null, so the fact that the distribution can be represented by a Gaussian distribution, you will find that probability of having women taller than 75 inches is virtually zero. It's 10 to the minus 10 or something. But in reality, there are some women taller than 75 inches. So you would find that the null model should be rejected. But still, I think we would agree that it's quite informative to represent, and it's a nice way to summarize this distribution, to have a Gaussian representation of the data. It's just that, okay, the extreme values cannot be represented by that model. Okay, so this is it for the basics of null model testings and a few already preliminary warnings of how to interpret or not interpret the results of null model testing outcomes. Let's now move to and describe null models of fMRI time series. What's the difference to example one with respect to examples 1 and 2 in the case of fMRI time series? Well, the only difference is that we are now in the multivariate case. So instead of having univariate metrics and models, we'll have multivariate metrics and models. So conceptually, it's going to be exactly the same as in example 1 and 2. It's just that it's going to be a bit more complex because we are in the multivariate case. So instead of variance, we'll have covariance. We have cross-correlation matrices, we'll have multivariate Gaussian distributions, etc. So let's take the simple example of the multivariate Gaussian null for centered fMRI time series. The null model in that case is that each time point, and again, each time point separately, has been generated by a Gaussian distribution with mu 0, because we assume centered fMRI time series, and covariance pattern or the correlation matrix sigma. So that tells you that's a matrix of size n by n, where n is the number of regions of interest. And that encodes the correlation or the covariance between each pair of regions between functional activity in each pair of regions of interest. Using this model, we can generate different sets of surrogate data. And again, each realization is now multivariate. The data being preserved are the mean and the covariance structure, and examples of tests would be, for example, the average lag autocorrelation over variables, because here we know that there is autocorrelation, and here autocorrelation, well, would be destroyed. So what is the expected outcome when comparing MO and MS? Will it be that MO is equal to MS because autocorrelation is preserved by M? Is it that MO is different because autocorrelation is not preserved by M and fMRI time series are known to be autocorrelated? Or will it be C, MO is equal to MS because fMRI time series are not autocorrelated? Again, take a few seconds to think about it. So I hope it's clear that it's not answer. Well, I hope. I think it's clear that it's not answer C because we know that there is autocorrelation in fMRI time series, right? So we know that there is autocorrelation here, but using this model that's that is IID, so independent for each time point is generated independently from the previous one, following this distribution. That means that here we won't have any autocorrelation. And since we know that there is autocorrelation in the original data, well, we expect to find M0 to be different from the autocorrelation M in surrogate data. So correct answer is answer B. So the conclusion of this is that of a potential test would be that if it's different in original and surrogate data, it means that data cannot be fully explained by the multivariate Gaussian. Again, relating this to the example of women distribution of women hate, this does not involve that sigma, the correlation is useless. I mean, it's not anymore to be proven that sigma is useful. If MO is equal to MS, there is no evidence that there is temporal structure in fMRI time series, but that's not what you would find, what we expect to find. Okay, so let's now move to another model, which is the first order autoregressive model of fMRI time series, which assumes that each time point is connected to the previous one through this autoregressive matrix A of size n by n plus Gaussian noise. So again, using this model, we generate surrogate data, multivariate surrogate data by inputting Gaussian noise here. And in this case, the data properties that are preserved are the mean and sigma as for the multivariate Gaussian, but also R1, which is the one lag autocorrelation matrix. So it's also n by n matrix, not symmetric anymore in this case. That encodes the link between variable I at time t and variable j at time t plus 1. So the diagonal would just be the diagonal of this matrix would just be the autocorrelation in all the ROIs, and the off-diagonal terms are called cross-correlation terms. What are examples of useful tests that we could have here? Well, first we could explore the amplitude of sliding window correlation fluctuations. That's what's being done in several studies. We could also compare the distribution of state transition patterns between original and surrogate data, etc. So just here to give an example of what we get if we compare the first metric, the amplitude of sliding window correlation. Here is what you get if you apply sliding window correlation on original data. So the blue curves here are the sliding window correlation for each pair of ROIs, and the red curve is the average of the blue curves. So as a function of time, you have quite large fluctuations, and this is what you get if you apply the same, if you compute the same metric in surrogate data. And in this case, what we showed in our 2017 paper is that it's much harder to reject, at least based on that metric, it's harder to reject the null, the first order autoregressive null, meaning that accounting for this R1 information actually allows to capture a lot of information included in fMRI time series. Let's now move to the third null model, which is phase randomization. I'm not going to enter the details of how that works, but essentially it's going from the temporal domain to the Fourier domain, the spectral domain, randomizing the phases, and then getting back to the original temporal domain. What are the data properties being preserved, and what is the model corresponding to this? So again, it's not that trivial as in the previous case. It's not that trivial to see that R1 is preserved. You have to rely on the Euler-Walker equations. Here, it's another set of mathematical properties that you have to exploit that are known as Wiener-Khinchin theorem. That allows to say that when you do this, you actually preserve the complete autocorrelation structure of the original data. So instead of just preserving mean, sigma, and R1, you preserve the complete autocorrelation structure, meaning the link between different time points, but at a horizon that is longer than just one time point. And the corresponding null model is the following: the fact, as I said, that different time points are connected, but with a horizon that is longer than one, up to t, the length of the time course. Again, we can have the same useful matrix M to be compared between original and surrogate. And the conclusion would be that I think, based on different results, that it's actually harder to reject this null model if you compare metrics in original and surrogate. So that was also shown in this paper by Hendricks. And the paper Pritchard and Taylor here is presenting the details of phase randomization. As a summary, here is the original properties of the data that are being preserved by the three different nulls that we've seen, so multivariate Gaussian, AR1, and phase randomization. You see that it's a nice nested hierarchical structure here for the properties that are being preserved. This one is obviously rejected because we know that fMRI time series are autocorrelated, and these two are a bit harder to reject. Okay, let's now come to how we should interpret significance of functional dynamics and how we should interpret outcomes of null models. And the question is essentially, do my results need to be different in original versus surrogate data in order for them to be relevant or interesting or meaningful? So by that, I mean, do I need, for example, a method to show, for example, if I have state transition between state 1 and 5, and I find that this is different in phase randomization and original data, does that mean that states transition, that this particular state's transition will be more relevant for brain fingerprinting? Well, the answer is just no. What null model testing allows us to do is to better understand the statistical information that is exploited by this state transition, but in no way it is informative about whether that particular metric will be more useful in a neuroscientific application, such as brain fingerprinting and things like that. So to better clarify this and maybe give a different perspective on this question, let's represent the overall statistical information contained in fMRI time series by this big green circle and the nested information that is preserved by the different nulls. So we have the Gaussian one, the AR1, and the phase randomized one that preserve increasing amount of information. Let's consider the case where I have a result, so a value of M that is the same in original and AR1 surrogate data. The result could be, for example, as I said, the transition, the probability transition between state 1 and state 3. We find that this is the same in original and AR1 surrogate data. Well, this just means that the method or the metric, in this case, the state transition probability between state 1 and state 3, relies and exploits statistical information that is contained within this this blue circle corresponding to the AR1 null model. So it could be, for example, exploiting the information included in this red ellipse. And again, in no way it's informative on whether this state transition would be more meaningful or more useful in clinical application. Consider another case where the result or a metric M again is different in original and phase randomized surrogate data. What curve corresponds to this case in your opinion? So again, take a few seconds while I'm checking and seeing that I'm running a bit late. Okay, so what is the information preserved by PR? Information is the light blue curve, right? We find the metric to be different in original and PR surrogate data. So this means that the metric is exploiting at least partly information that goes beyond this circle, right? And so the correct answer is curve 1, because it's the only one that exploits information that is here contained in this part of the remaining original statistical properties represented in green and beyond the PR statistical information. It does not mean that it's only exploiting this. It means that it's at least partly exploiting nonlinear or non-Gaussian features, as the PR null can be framed as preserving linear and Gaussian features. Okay, so this is essentially what I just said. And so I wanted to end up with a few remarks. So the first one is that sometimes it is claimed that we test against surrogate data, for example, phase randomized surrogates, to test whether the result is trivial and explained by a trivial model. So it's true that to some extent, the phase randomized null corresponds to a linear and a Gaussian model. So in that sense, it could be called trivial. But if you think in terms of parameter complexity, phase randomization, as we saw a few slides ago, corresponds to a linear model and preserves the complete autocorrelation structure. These are all n by n matrices, and we have t plus one of them. So we are more or less approximately preserving t times n squared parameters, which can be huge if we have t, for example, on the order of hundreds, and t 100 to thousands, and n more on the order of a few hundreds. So this is huge, and I'm not sure we should claim this as being trivial. So it's trivial because it's linear and Gaussian, yes, but in terms of parameter complexity is not that trivial. So I'm not sure this holds as an argument or as a motivation for doing surrogate testing. Then which proportion of statistical information is captured by these models? So in other words, are we in the case of the left figure, or are we in the case of the right figure? My impression, but I might be wrong, my impression is that we are more in the case of the left figure, because converging results tend to show that it's hard, quite hard, it's not impossible at all, but quite hard to reject phase randomized null, so the one that is represented here, or even the autoregressive null that is represented by this intermediate blue circle. So since it's quite hard to reject the null, my impression is that these models, linear and Gaussian models, are capturing quite a lot of information that is represented, that is encoded in the overall fMRI data sets, instead of this, where, well, it would be very easy to reject the phase randomized, which is not what we observe in fMRI time series. And then an even more important question, and I think even more tricky question, is to know where the interesting information is. So first we have to define what is meant by interesting, but for example, it would be what we want eventually to do is to have a good classifier, to have a good marker for brain fingerprinting or for early disease diagnostic. So where is this interesting information? Is it in the more central parts here? Is it in the outer parts here? Well, it's not anymore to be proven that the properties preserved by the multivariate Gaussian, for example, the correlation matrix, is very informative and useful in these applications. And for example, brain fingerprinting is essentially based on these metrics and is doing quite well, very well. So do we want to go and explore these nonlinear and non-Gaussian features here, which come with much more complex models, obviously, because they are nonlinear and non-Gaussian? I'm not sure. Maybe we should focus more on exploring the information that is contained in these next circles, so contained in autocorrelation structure, for example, and that were shown to represent and capture quite a lot of information that is contained in fMRI time series. But again, this is an open question and I have no definite answer for this. So the take-home messages would be the following. First, and I would say most importantly, null model testing just and only allows to better understand and characterize the statistical information that you have in fMRI time series, or the statistical information that a given metric, for example, a state transition between state 1 and state 3 again. So to better characterize the statistical information that metric exploits. But it should not be used and seen as a mandatory validation step to assess relevance of a metric, for example, for fingerprinting and for classification. So framed differently and maybe a bit in a provocative way, I have the impression that in most cases, and most of us would not be interested in what null models can tell us about the data and about the metric. So maybe, yeah, think twice on the relevance of doing these tests. If you are really interested in knowing the nature of the statistical information exploited by your metric, then you can use null models. Otherwise, if you just want to show that your metric is relevant for brain fingerprinting, but well, then assess robustness of brain fingerprinting, but there is no real relevance to compare it against brain fingerprinting achieved in a surrogate dataset, meaning a surrogate of the fMRI time series. Then the second point, which is that rejecting a null should not be the end goal of everything. I mean, you will always find a metric that, for example, exploits extreme values of a distribution that allows to reject the null. So this should be more thought in a broader context, where you think in terms of benefits and of going for a more complex of a more complex model. So rejecting the null essentially does not discard the use of this null model. And the best example for this is the use of the correlation matrix that is preserved by a null, the multivariate Gaussian, that is very easily rejected for fMRI time series. Then finally, if you want to use models that will not be trivially rejected in fMRI time series, we suggest to use AR1, so multivariate autoregressive models of order 1, or phase randomization. I think that you will find that it's not that easy to reject a null, and it's fine. I mean, that does not discard your metric or anything. It just tells you that your metric is exploiting linear Gaussian properties. Here are the references. So the first one, where we detail and we go in much more details, we also present alternative models, and we discuss other null models that have been recently used in assessing and exploring dynamical properties of fMRI time series. So I think AR1 and PR are the most important ones. We discussed briefly other methods there, and we give also a few examples, more examples, concrete examples based on fMRI applications, and we discuss this. Pritchard and Taylor paper is the paper where they present phase randomization in particular, but they also have a very extensive description of many different null models and the link between the null models. So it's very interesting if you want to well, draw links between these null models. Here, the paper by Hendricks that, as our paper, show that it's not that easy to reject the null, essentially. And if you want to know more about modeling and understanding the properties preserved by phase randomized AR1, etc., we detail that in our 2017 paper. So with this, I thank you for your attention. I hope you enjoyed the presentation, and I look forward to have a chat with you in the interactive session. Thank you.
Good morning, good afternoon, wherever you may be. Good night. I'm going to be talking about time-varying connectivity in the context of data-driven approaches and focusing on also applications to clinical populations. I'll cover 5 main categories here, and we'll start with just a brief introduction. In terms of history, in 2010, a couple papers came out that introduced sliding window approaches focused on either time frequency or whole brain ICA dynamics. And that's really when I think whole brain dynamic connectivity sort of began. I distinguish that when I say whole brain from other methods like dynamic causal modeling and lots of fMRI methods that look essentially at dynamics of signals. Here we're talking about dynamics of, sort of interactions between large brain regions, large sets of brain regions. That was followed shortly in 2011 by another sliding window approach, but looking at spatial aspects of the default mode network. In 2012 and following is where things I think really began to take off. Elena Allen's paper focused on sort of identifying states of connectivity, transient periods of reorganization. Jones and Damaraju and several others started to introduce clinical applications, looking at Alzheimer's and epilepsy and schizophrenia. There are a number of reviews these days. You can see 6 of them listed down there. Matt Hutchison's paper was probably the first comprehensive summary. In 2014, my review introduced the term connectome, just another yet abuse of the omic notation. And then there's a paper by Dan Lurie there that I'll just highlight, which gives a real extensive coverage of many, many issues in the field. Finally, Armin Araji's paper covers spatial dynamics. And I'll cover that a little bit later in the talk. Now, if I just introduce for the moment now what I'm going to call the windowed FNC approach. And so here we're focusing primarily on time courses that come from ICA components. Okay, so their cross correlation or temporal dependence among those is called functional network connectivity. And so if we take those and instead of looking at the average over all time, we do sliding window approach to get essentially variations in connectivity. And then we can look at clustering of those into states that may or may not be overlapping depending on how you want to model it. Essentially, we're interested in looking not at the static connectivity. Here it would be a correlation of 0.4, but we want to see these transient strong and weak coupling that may occur, which we call temporal dynamics. And so here we have a correlation of 0.8 and 0.1. Here's an example looking at a study of schizophrenia versus healthy controls. And we essentially used dynamic connectivity states estimated from the controls and from the patients. And if you'll focus on here, the bottom row where we're looking at the difference between the two, you can see that there are sort of distinct regions showing differences in connectivity in the different states. So for example, these 2 states are showing strong differences in primarily cerebellar and subcortical regions in interactions with the sensory motor. Once we have these states characterized or estimated, we can compute metrics from them. For example, dwell time, occupants, frequency of occupancy. We can start to look more explicitly at dynamic relationships. But if we simply look at mean dwell time, which is the amount of time that a state that an individual stays close to one of these states after it enters that state, or after it gets close to that state, we see that healthy control shows significantly more dwell time in state 1 and state 2, whereas the schizophrenia patients spend much more time in those sort of weakly coupled state 4 and state 5. So just one example. And we can also characterize and come up with different metrics to sort of summarize what we see in these states. One of those is called metastates, and I think this term is used a little differently in different papers, but in this Miller et al. Paper, the way it was used was, so we have 2 different states here, one versus the other, and each subject starts somewhere in this, grid at time 0 and then moves around, throughout the experiment. Another individual may have this kind of a pattern. So every point in time can be characterized as a 2-element vector where the number of elements is equal to the number of states. And then the number tells you the degree to which they are strongly positive, strongly negative, or near 0. And so each one of these vectors here is called a metastate. We can then summarize metastates with a number of metrics. For example, the number of distinct states, the number of changes, the total distance traveled in that state space. And so, that gives us some really interesting sort of global summary metrics of dynamics. And here, for example, you can see what was interesting was, regardless of how we estimated the states using either ICA or PCA or k-means, we see comparisons between schizophrenia patients and healthy controls show similar directionality in that they showed reduced dynamic fluidity, reduced dynamic range. And you can compute other sort of more interesting metrics. Here's another one that Robin Miller developed called the concept of a hub state, which is the state that, essentially when you enter that state, you tend to not leave it. It's sort of like a sticky type state. And she showed that occupancy in hub states was correlated with negative symptoms in the context of schizophrenia patients. And there's also been a number of studies starting to look at the relationship between dynamics of activity and dynamics of connectivity. So linking of essentially first and second order dynamics. Here's one example where we looked at dynamics of the ALF metric, which is sort of amplitude of low frequency fluctuation, or the spectrum, and the dynamics of the functional network connectivity. And they both showed evidence of considerable dynamics that correlated, in this case, with various measures related to schizophrenia patients versus healthy controls. And here you can see, for example, the FNC states, and here you can see the ALF states. There are 6 of them in this case. Now, something that I think is important for the field is to really think about, the terminology that we're using. And I think most of the early papers are focused on what I will call temporal dynamic measures, which is essentially you have fixed nodes and you're looking at changes in the coupling between the nodes. And yes, that can manifest as sort of a spatial pattern, but it doesn't directly tap into aspects of spatial dynamics. A spatial dynamic measure is where the nodes are flexible, where they're moving around or shrinking or growing, for example. And spatial temporal dynamic is where both the coupling and the nodes are changeable, or maybe changeable. And so here's just an example of, one way of getting at that, which is essentially treating it as a hierarchy, where the larger hierarchy level consists of a number of sort of basis sets that as they are weighted differently, they manifest as different dynamic patterns over time. And so here you can see, for example, for the subcortical region, you can see that changes that are occurring over time. And if you characterize these, you see differences in various clinical data sets, quite substantial differences in them. Now, this is really just looking at sort of granularity, but we can also look more directly at fluid dynamics, or sort of allowing the networks to sort of vary voxel-wise, whatever the finest grain scale of measurement that you've collected. And so here we have, an example of this from Armin Raji, where essentially looking in this figure here in the bottom left, we have default mode network and salience network, and you can see white regions where they overlap for pretty much all of time. And then where you have colors, you see that the portions of that network spend more time in different states. They come in at different times versus the other regions. So a blue region and a green region and a red region occur at different times. We can also characterize these spatial dynamics using additional metrics like a spatial histogram, for example, here showing a default mode network. And this is again showing differences between patients and controls. For more details, you can look at this paper here. So this again is just a sort of teaser. It's basically saying let's look at the different approaches that are out there, look at how they are actually, what they're actually measuring. And is it temporal dynamic? Is it spatial dynamic? Is it both? Pretty much any method can be adapted or extended to be any of these, but really the concept is, are we talking about the same thing? And so I think that's important to keep clear. Now, a few slides on sort of validation. I think, there's been a lot of studies now, but some early ones that we did looking at replicability. So we took 7,000 subjects, divided them into sets, 28 sets of 250 subjects, and we compared, for example, how consistent were the dynamics that we estimated. And the explicitly dynamic metrics, like the metastate metrics, for example, were quite consistent across independent sets of subjects. And if we perturb the dynamics using, phase randomization, for example, it completely breaks down the dynamics as expected. There have been a few multimodal studies. Here's one looking at EEG and fMRI collected concurrently. And you can show that if you measure sort of drowsiness aspects from the EEG data, you can see that it correlates with patterns in the fMRI going from sort of anti-correlations present in the least drowsy state to virtual hypersynchronization in the most drowsy state. And these are also predictive. You can predict sleep. So Enzo Tagliazucci's group had a number of studies where they looked at predicting sleep stage. This is one where we worked with some of their data, and we showed that we can predict sleep stage using these state-based approaches quite consistently and quite accurately. Interestingly, you can also predict you can classify particular states within sleep conditions as well. And so that's here showing classification of wakeful states, again, with about 90% accuracy. It's a small study, but again, it's intriguing, I think. And whole brain connectivity is dynamically task dependent. So this is data collected by Javier Gonzalez Castillo's group, and we're basically showing that if you apply this state-based dynamic connectivity approach, you essentially map the tasks for these sort of pretty intensive tasks, ranging from rest, memory, video, and math, and you can get a pretty clear difference. Basically, you just cluster them into the tasks. But then it gets more interesting if you start to look at within tasks. Can you look at combinations of these states? And this is some work from Jason Nomi and Lucina Uden, where they showed that in the human connectome data, the dynamic FNC properties are related to executive function pretty explicitly, and those replicate across independent data sets. And the last sort of one I'll mention is that obviously there's a lot of simulations out there. It's important to be careful about how you define what is a null model in the context of dynamics, because if you show, oh, things are different during time A and time B, that's not evidence that there aren't dynamics in there. That's really not a good null model for dynamics. So again, these are sort of simple things, but it is important to keep in mind. We looked, for example, at a measurement of kurtosis versus something we called a multivariate phi measure. And kurtosis is very sensitive to outliers, to things like movement, but it's not a particularly good measure of dynamics, whereas this sort of wavelet-based multivariate phi measure is indeed quite sensitive. And I would also sort of point out it's important to keep in mind the terms stationary and non-stationary have explicit ensemble-based meanings, and you can actually still have quite a lot of interesting dynamics even if the data are by definition stationary. So it's important again to keep that in mind. So now let me just run through a few clinical applications just to kind of show you what we find. Here is looking along sort of in a mood psychosis data set. We showed that you see quite significant differences between healthy control, bipolar, schizoaffective, and schizophrenia individuals in the different states, and the patterns of connectivity are mapping quite distinctly onto these different states and representing different patterns that are sort of characteristic of these disorders, suggesting this might be meaningful or useful as a way to characterize or identify or summarize mental disorder. Interestingly, though, it depends on how you look at it. So this is some data looking at ADHD with Nina DeLacy. And what she found was, she sees reduced dynamism if she looks at essentially a global level. So things like metastates, at a local level, she sees reduced dynamism, and at a global level, she sees kind of a mixture of increases and decreases. So depending on the level at which you look at it, whether you're looking within a network, whole brain, or sort of globally across all the states within the data set, you're going to see different patterns of connectivity. But it gives us also sort of some hope that we can start to compare and understand a little bit brain disorder. And so here is an example where we used a fully automated ICA approach called NeuroMark, where we're basically taking templates derived from large data sets and using a spatially constrained ICA on individual subjects. We have comparable then components that we can compare. And so here you can see on the left, schizophrenia, on the right, autism spectrum disorder. Here we're looking at differences in something called stepwise functional network reconfiguration. This is some work by Zhenning Fu. That's essentially a measure of change step by step in the network parameters, in the windowed parameters. And we see sort of commonality in the SFNR. You can see a reduction in the patient group, in the control group relative to the patient group. You can also see that schizophrenia exhibits strong abnormalities along these lines in the cerebellum, and in the subcortical, where these stars are significant. And then thirdly, you see ASD is a little bit more spread across domains, and also not quite as strongly significant. And then in this third point here, we looked at the overlap between cerebellar and sensory motor domains, where there was overlap. So you can see here and here. And we see that it was correlated with reasoning problem solving and autism diagnostic observation schedule within both schizophrenia and autism spectrum. We saw a relationship between those two. So again, it gets at distinct, disorder specific aspects, very complex. There are a lot of studies trying to look at how to characterize mental disorders. And so this, I think, gives us some promise going forward. And we can also use dynamic FNC to look at subgroups. And so here's just one example looking at subcortical ischemic vascular disorder and autism disease. And so we found significant differences in the dynamic parameters, looking again at dwell time in the different states between sort of this vascular type of AD and AD itself. And here's another study looking at substance use. And this is looking at multiple types of substances. So we had several hundred data sets. We're looking at alcohol, smoking, combinations of alcohol and smoking, marijuana, etc. And we see the dynamics sort of give you unique patterns of differences, where you see smoking kind of more broadly hits multiple states. And you can see state 5 is the most strongly impacted by multiple substances. And then lastly, if we're interested in doing predictions, so cross-validated prediction, then dynamic connectivity appears to be more predictive than static connectivity. And you might assume that because, in resting fMRI data, things are changing, they're not very well controlled, but if we can characterize their dynamics and the parameters of their dynamics, that might be a more sensitive measure. And that does appear to be the case. Here's a direct comparison of static FNC and dynamic FNC. And if we classify with just the static, we get about a 60% accuracy for comparing 3 ways, bipolar, schizophrenia, healthy controls. Dynamic is about 84, combined is about 90%. We also looked at prediction of mild TBI, which is very challenging because it's very subtle and very heterogeneous. And we find that if we compare, resting fMRI generally did a little bit better than diffusion-based measures, at least in our hands. And we also see that the dynamic rest fMRI did better than the static fMRI measure at prediction. And we've also looked at, can we look at the number of states? How do we set the number of states, right? So that's a free parameter. One way to do it is to use prediction accuracy. And so what we do find quite a bit of variation. Here you can see comparing a prediction of schizophrenia versus controls. We see static, and then we see the accuracy jumps up if you go from 1 to 2 states, and then it varies a little bit among, the accuracy varies a little bit, going from 2 to 10 states, for example. We have a peak at about 4, and if we these are sort of cross-validated results, so these are looking at, once we identify the most predictive number of states, then we go to unseen data and we perform a test prediction. We also have metrics that we can use. There's been a lot of work looking at the elbow criteria and other things. What we showed in this paper, again, the Vergara paper here, we looked at 24 different indices, including that one. And we found that the Davies-Bolden and the Ray-Turi index appeared to be the most suitable, the most consistently correct compared to the ground truth. In terms of a synthesis, we're still working on this, and there's still a lot more studies need to be done, but there do appear to be some disorder-specific patterns of connectivity that we see. We see consistently, for example, that schizophrenia shows the most difference, the reduction generally in all sorts of dynamism type measures. And you can see some of the sort of observations that we're showing here, where down is decreased, up is increased, and the number of arrows tells you the sort of magnitude of the effect. In terms of advanced metrics, I'm just going to go through a few of these just so you kind of have a sense of what's out there. There's still a lot of work to be done in this field. So if you're getting into this, learning about this here in this course, we hope that you'll start to develop some new techniques. But here's one where we were looking at time frequency analysis. So we basically can estimate states in a way that's sensitive to using a wavelet-based approach. Not only the magnitude of the correlation, but also its phase. Is it correlated or strongly anti-correlated? That's the color. And also the spectra here. You can see, does it have a peak in this sort of mid-range, so the bold frequency range, or is it low frequency? Is it high frequency? And so this, we showed, is able to separate states that may be overlapping in pattern, but have distinct frequency profiles, for example. There's also a number of approaches to try to, address some of the limitations in using a sliding window. You can use instantaneous approaches. Here's one which we call the windowless approach, where essentially we don't want to blur together if there's sort of a rapid change and you have a sliding window that goes over that. It can sort of artifactually present a third state when there isn't really one there, right? And so we showed that you can use this sort of instantaneous approach based on KSVD to directly estimate the states. And we found the good news was that the states look pretty similar to what we found with the windowed approach, with the exception of the sort of least, the one that looked sort of weakly modularized in the sliding window approach had more strong modularity in the windowless approach because it's presumably more transient, getting blurred out. You can look more generally at sort of a more unified approach. This is Ashkan Fagiri's work, looking at a filter bank. So essentially we cover all the different frequency ranges. And we directly, sort of have a model that goes from activity all the way to connectivity across multiple frequency ranges. And so then we directly estimate, the amplitude of the frequency in each band, as well as the states. And so we showed, for example, that we can, we can accurately capture information both at low frequency and at high frequency, whereas the window, which is a choice of filter in itself, can only capture information within that range. There are density approaches out there, a number of them. This is one we called, brain trajectory dynamics. So essentially, you're looking at say this is one subject moving around in time between two states. You're looking for sort of dense regions and sort of represent those as sort of a pattern, a spatial pattern. So again, looking at the trajectories a little more flexibly among them. If you focus on a state-based approach, you can look at modeling those a little bit more interestingly using an attractor model. So here what we did was we instead of using the amplitude only, we took so we have this sliding window connectivity patterns, but we also have the derivative or the change over time of those patterns. And so that derivative then tells us whether information is kind of flowing into or out of, for example, the pattern at time point 1. And so what we found was when we did the clustering on all of these, both the dynamic and the both the 0th and the first order derivative, we find these pairs in some cases of states where the derivative was showing kind of the opposite effect, the opposite sign, whereas the amplitude itself was very, very similar. You can model that again as sort of a set of attractors that sort of move around relative to one another and grow closer and farther, sort of in an oscillatory type network. We looked at this in the context of TBI, and we found pretty interestingly, Hans van der Hoorn showed that these attractors get disrupted briefly after injury, partially recover, and then in a longitudinal study 3 or 4 months after have shown that showed that they were fully recovered. You can also start to search the dynamic space a little bit more. So again, this is very high dimensional data. It's always a problem that we have. But this is just sort of a quick sampling, a quick teaser of, we can cluster, we can look across, the entire data set to find time points that are similar, pairs, connectivity values that are similar, subjects that are similar, and essentially try to compare apples to apples, right? So we kind of divide the data into these subsets. So for example, here in color, we see regions that we see basically about 3,500 windows, 81 subjects, 619 pairs that showed similarity to one another. They were close to one another in their mean values. And when we do this, we find that we get much more sensitive relationship to things like symptoms of the patients. Here's another work from Apo Hyvernin's group looking at using a deep learning approach to look at temporal primitives across the data, essentially finding these little snippets of time, sort of chirps that show up in the data, and looking at their relationship to various, tasks and behaviors. And lastly, blending what I like to work with ICA-based approaches because it gives us a lot of intuition. We get these networks, we can help interpret those networks. But we can also benefit from deep learning by which gives us potentially increase in flexibility by blending the two. And so here what we did was we took ICA networks as priors, estimated using a deep residual network, a full 4-dimensional pattern of change over time for each of the networks. So for example, if we didn't have enough data already, the default mode network then becomes a 4-dimensional network over time. And we have that for all of the networks. So we have a 5-dimensional data set, and then we can then take those and then reduce them back down into more intuitive summaries. For example, the average within the network. We can look at the relationship of variation over time to things like age. If we look at the variability in the amplitude versus the change over time, you can see that it's quite striking that we see, for example, in the sensory motor cortex, the amplitude highlights just this portion, whereas we get these regions showing strong dynamic behavior, but they get averaged out, blurred out in the static method. Sorry, that's not even static, that's the average over, over the dynamics in the amplitude. And this is just sort of the last slide I think I've got, which is basically to show that, now that we're getting more comfortable in, that these dynamic connectivity approaches are meaningful, we're also starting to look at linking them to other kinds of data. This is one example I like because we looked at single nucleotide polymorphism data, and we essentially showed that from that early slide I showed you in schizophrenia patients, we have these states, right? And we showed that schizophrenia patients were less present in these modular states. The healthy controls spent more time in the modular states. we looked at the relationship of these to the SNP data and using an approach called parallel ICA. And we essentially found a linked SNP component that was strongly anti-correlated with a risk score, a polygenic risk score for schizophrenia. And interestingly, if you have a higher risk for schizophrenia, you spend less time in these modular states. So it's sort of directly, it's consistent with this risk score measure of schizophrenia. It's consistent with the work that we looked at earlier on, which showed the schizophrenia patients do indeed spend less time in those modularized states. So now for software, we've developed a number of tools based on ICA. So we have this group ICA of fMRI toolbox called GIFT. It has within it a dynamic functional network connectivity toolbox. I'd recommend this paper here where we actually cover a review. We do a review of dynamic connectivity in general. We're not limiting it to just the data-driven approaches. We're covering a lot of different approaches. And also here's some videos where we introduce this dynamic FNC toolbox and dynamic FC toolbox. So essentially you can use it to analyze data coming out of ICA, but you can also use it to analyze data coming from other approaches. So it's not limited. You can use voxel-based approaches, region of interest-based approaches, or ICA-based approaches in the tools. So I would recommend that if you want to just play around and see if it's useful. Please let us know if you have any questions about it. Here's just a few videos, sort of pictures, snapshots rather, of the output from that tool. And with that, I just want to say thanks to the folks at Trends who have done a lot of this work. Thanks to the funders, and thanks to you for bearing with me, and hopefully we can have some good discussions.
Hello from Portugal. My name is Joan Cabral, and I'm going to talk about mechanistic principles of resting state networks and their modulation over time. So briefly, I'm going to go over some empirical evidence of resting state networks and discuss on these conflicting mechanistic hypotheses. And then I will show some results, some insights I've been gaining with the leading eigenvector dynamics analysis into the temporal signature of these resting state networks, and also how they can be modulated by different psychoactive drugs, such as psilocybin and DMT. So spontaneous functional connectivity, as you probably all know, is characterized by these long-distance correlations between brain areas that are typically functionally related. And so, in other words, these consist of voxels that share a common temporal signature. So this is what underlies functional connectivity. So across fMRI studies of spontaneous or resting state brain activity, there is some consistency in the detection of so-called resting state networks, also called intrinsic connectivity networks, that in a way consist in subsystems of the brain, so groups of brain areas that engage together, exhibiting a characteristic temporal signature, in order to somehow integrate information, so their function is still unclear in the resting state. But here what I want to show is that irrespective of the methodology, so you can either use ICA or clustering or seed connectivity, there seem to be a discrete number of patterns that seem to be related to information processing in the brain. Now, the thing is that these spatial patterns or the characterization of these subsystems depends a lot on the methodology. So there is little consistency across studies, or there are a lot of different ways to capture these resting state networks, let's say, and it's hard to tell which is the best. I mean, all of them are good in their own way. And then there is this issue of the unclear number of resting state networks. So overall, people agree that they are discrete states, so they are different subsystems, but the exact number is undefined. And as well, there is this issue regarding, some studies consider that the same area of the brain can belong to more than one resting state networks, so there is no exclusivity, so they allow spatial overlap, whereas other methods define these intrinsic connectivity networks as spatially independent, so the same voxel cannot belong, or the same brain area cannot belong to two networks at the same time. So there are a bit these inconsistencies, but overall I think they all point to this consistent existence of resting state networks. Now, one thing that to me becomes really convincing of the importance of these subsystems is their relevance for psychiatry. Over the last decades, there have been a lot of studies showing that the integrity of these different types of resting state networks or intrinsic connectivity networks appear disrupted in some psychiatric disorders. So the thing is, although there is this nice review from Leanne Williams from 2016 where she tries to put together the different evidence from different studies, and overall she comes up to different subsystems. So she characterizes these different networks, not just the default mode that is the most well known, but also the salience network, another network that is related to negative affect, positive affect, attention, and cognitive control. And so they nicely link to the patterns that are found in the brain at rest, not directly, but with some overlap. And the question is, if we understand better how these patterns are generated, where they come from, we may be able to help in the rebalancing of these networks that seem to be disrupted in psychiatry. So the main question that has been puzzling me over the last years is, what are the generative mechanisms of resting state networks? What couples brain areas together in the spontaneous state? So we all agree there is a strong role of the structural connectivity, of course, yes, that is the most direct answer. What couples brain areas together? An anatomical link, yes, but why do they spontaneously engage in transiently correlated activity? And to understand that, we would need to have a better insight into the temporal signature of these networks. What I mean is, do they just simply coactivate, so the brain areas that belong to the same subsystem all exhibit typical canonical hemodynamic response function, so they all activate and then deactivate together, maybe with a certain phase lag? Or do they engage in a synchronized behavior, so transient synchronization, that is, they exhibit sustained periodicity in their shared temporal signature? And although these two mechanisms can lead to correlations like functional connectivity, dynamic functional connectivity, the mechanistic generation can be explained by completely different fundamental principles. And so this is why this is important to gain insight in this direction, to disambiguate these questions in order to gain more insights. And so one way to explore synchronization mechanisms from resting state fMRI activity is to take the signal in a given parcellation scheme and then compute the Hilbert transform to extract the signal phase, the phase of the signal. So it's a complex signal, and then we can compute the synchronization matrix, which tells us at each instant of time how the phases of the signals in area A and B are aligned. But although this matrix looks very rich and dynamic, it is very noisy and very hard to analyze. Importantly, it's nice to see that we see some recurrent patterns that seem to appear over time, so there is some structure underlying it. But if we take the leading eigenvector of these matrices at each instant of time, we can see that the patterns that we observe in the synchronization matrix are characterized by different subsystems that consistently engage in phase or in anti-phase. So one way to characterize the non-trivial organization of phase synchronization across brain areas is to compute this leading eigenvector of the fMRI signal phases. To try to give an intuition of this rather abstract concept, we can imagine that the leading eigenvector is as if we would draw an arrow that crosses the brain that captures the main orientation of signal phases in the brain. And so some brain areas would project in one direction of this orientation, so here they would be colored in blue, and other brain areas would project in other direction, like here they are colored in red. And of course, if all the brain areas are going in the same direction, there would be just one color. So that is what I call the global signal, so we will see. But above all, this is what it does. It just captures the main orientation and then sees how the different brain areas project into that. So this is what we are seeing, and it's represented here by this arrow. So the thing is, if we cluster these leading eigenvectors that we obtain at each instant of time using a given, like here we use the k-means algorithm, we find that we can detect centroids of this cluster that overlap with the functional networks or the resting state networks that are reported in the literature. So this appears to be one more way to detect resting state networks. Now, one of the advantages of this method is that the k-means then assigns to each TR, so to each volume, a centroid, so one of the states. So it will tell us for each TR, we can say, okay, which was the resting state network that was dominant at that instant of time. And then taking the time points that are assigned to a given pattern. So for example, if we take all the time points from all the scans and we go back to voxel space, so taking the volumes in MNI space, we can then average the fMRI signals that were assigned to a given state to see, is this really a coactivation? And if we do this, we can see anything, like, so in each column here, so this would be the mean volumes representing state number 1, state number 2. There is here maybe some more red, so higher fMRI signals, lower. But overall, this seems very, very blurry. Now, the thing is that if instead of averaging the fMRI signals in the voxel space, if we instead calculate the leading eigenvector in voxel space and average these leading eigenvectors, what we detect are clear definitions of these networks in voxel space. So this would be the global state where all voxels are aligned in the same direction. So in this case, all in blue, except here from some strong phase-locked activity in the trunk here, in the close to the fourth ventricle. Then in state number 2, we clearly see the default mode network. State 3 here is more the limbic system. State 4 here is the visual. State 5 is the frontoparietal. And state 6 is the somatomotor network. So there seems to be some much better detection of the states or of the functional networks if instead of taking the mean activity, we take the way their phases are locked in space. So in a way, we can reject the hypothesis that the activation of these patterns is related to just co-activation. So it's something that is actually rotating, but it's still unclear why this method is working so nicely and revealing such clear patterns. So the thing is that we are finding these phase eigenconvectors, what they are capturing is phase relationships between brain areas. So it doesn't matter. What matters is that when one goes up, the others go down, and vice versa. So it doesn't matter which one is going up. So in a way, it reminds us the behavior of standing waves in bounded systems, where there are patterns. So here for each of these waves, there would be one eigenvector, and then this eigenvector can oscillate in time. So this to convey this. So now that I described some insights regarding the nature of resting state networks, I'd like to come back to this work by Lina Williams, and where she, beyond identifying these different subsystems or subnetworks that are important for psychiatry, she does this speculative connection of these different networks with ways of intervention, so how we could modulate these states. And it's interesting because the idea is that, for example, the default mode network could be modulated both with mindfulness but also with transcranial magnetic stimulation. On the other hand, the salience network could be modulated by SSRIs like these serotonin reuptake inhibitors, but also by deep brain stimulation. So there seem to be different ways to tune these networks, these patterns, that can be natural, like mindfulness, cognitive behavioral therapies, or pharmacological, with the use of drugs, or by electromagnetic perturbation. So there are these different types of ways to perturb these systems that are important to also take into account, because they may reveal also some insights into what is actually going on. So one of the studies where we studied indeed the effect of a given drug, a psychoactive drug, on the modulation of resting state networks was studying the effects of psilocybin, which is the psychoactive compound in magic mushrooms. And briefly, the participants were scanned before, like for 5 minutes before being infused with psilocybin. Then there is a minute where psilocybin is infused inside the scanner, and they are scanned again. And then 2 weeks apart, they were scanned. The same procedure was repeated, but with a placebo. And so here what I show is that there are the fMRI signals in 90 brain areas, and the color shaded bars at the background correspond to the cluster assignment that is returned by the k-means, so which means that each different color corresponds to a different brain network configuration. Now what is nice is that if we calculate the probability of dominance of each of these networks, so how the k-means assigns each state the probability of assignment to each of these states, we find that there is no significant differences before and after placebo infusion. Even 2 weeks apart, before the psilocybin infusion, we also did not detect any significant differences, but once psilocybin was infused, it had a strong effect, particularly in this network that has been related to cognitive control. So we can find that there is a given molecule that is acting specifically on one particular functional network. Now, when we applied the same approach recently, we've been analyzing data using DMT, which is another psychoactive compound that can have some similar effects to psilocybin, but not all. And what we found is that if we apply again LIDA, we detect here they are represented in a different way, but they in a way return the same pattern. So this would be the global mode, the default mode network, the frontoparietal, the visual, the somatomotor, and here the limbic. And here I'm coloring or rendering the AAL brain areas that have their signal shifted in this leading eigenvector, right, from the rest. So all these areas are synchronized in phase, but in out of phase with the ones in transparent. Okay. So below I show the matrix pattern that each of these networks would give, but importantly, at each instant of time, it's a sum of these different patterns, right? So we would have the instantaneous connectivity matrix would be a weighted sum of these different patterns. Now the k-means sees which one is dominating in variance, so takes the one that dominates. And we find that with DMT, so here the probabilities of occurrence or of dominance are shown at the bottom, and the last condition, it's quite small, but you can trust me, the last condition is the one where DMT was infused. So this would be before placebo, after placebo, before DMT, after DMT. And we find there is an increase of the global signal, let's say, and a significant decrease, so not just of the frontoparietal like we had seen with psilocybin, but also of the default mode networks. So these two networks that are associated with higher order cognitive functions seem to be strongly suppressed with DMT compared to the other conditions. And we find also an increase in the limbic system. So there is an increased probability of the dominance of the limbic system. So this gives again a very interesting insight that these molecules don't act in the whole brain activity in all the subsystems. They act on specific subsystems. And now analyzing the temporal activity of these different networks. So let me explain briefly what this figure represents. So for each of these 4 blocks is one of the sessions. So this is before placebo infusion, after placebo infusion, before DMT infusion, and after DMT infusion. And each of the lines corresponds to the cluster time series, so the occurrence of each of these patterns for each of the subjects. So if we have 20 subjects, for example, subject 1 in the first TRs is always in the global state, then switches to the frontoparietal, then to the limbic, then to the global state, and so on. So here is the legend of the colors. And if we look overall to these patterns, we see that like the distribution of colors, let's say, seems quite consistent across these 3 conditions, non-DMT. And as DMT is infused, indeed we find more green, particularly in the beginning, right after the infusion. So this is the first, these are divided into minutes, let's say. So these white lines define different minutes, and in the first minute we clearly see there is much more green, indicative of the occurrence of the limbic system, and much less orange and yellow as we would find in the other patterns. So below is for each minute a pie plot indicating the probability of detection across subjects, across all the subjects in that first minute, the probability of each of the patterns. And so indeed it's nice to see that it's not just a change at the block level, right, at the whole 5-minute scan, but we can really see that in the first minute the occurrence of the limbic increases up to 25% compared to around 10 in all the other sessions, and slowly decreases also over time. And simultaneously, the DMN and the default mode network and the frontoparietal that are significantly suppressed start increasing again over time. And so, in a way, DMT is nice because it has a very rapid action, so it's not a long-lasting effect. And so we can capture the whole experience inside the scan. and it's revealing some consistency. So this work is still in preparation. I hope to put it out soon, but I wanted to share because I thought it was relevant for this workshop. So overall, I think, well, we all agree that the biophysical mechanisms underpinning functional brain networks remain unclear. Our results indicate or suggest that resting state networks may oscillate in time. So they may not be just characterized by co-activations, but by oscillations, so synchronization mechanisms, and that they are modulated by psychoactive molecules. But this isn't new, but at least we find this specificity. Importantly, we suggest that further insights into the temporal signature of functional networks is needed to gain a better understanding into the physiology of these networks and ultimately into psychophysiology. So what are the mechanisms, the physical mechanisms, the science behind the psyche. So to help as well in psychiatry. So overall, I'd like to thank you for your attention. I am based at Nuno Sousa's lab in Portugal, but I also keep strong collaborations with the group of Morten Kringelbach in the UK and Denmark, with Gustavo Deco in Spain, and in Lisbon with Noam Shamesh. So if you want codes, or I have a GitHub code repository, but I'm leaving here my email because it's nicer to have the latest updated codes. So let me know if you have any doubts or if you want help to apply these codes to your data sets, or also to help me gain better insights into these mechanisms. Thank you for listening. Bye.
Hello. I'm Michael Breakspear. I'm a professor of neuroscience at the University of Newcastle in Australia. And I'd like to thank the program committee of OHBM for selecting our educational course on time-varying functional connectivity for this year's meeting. I'll be spending a few minutes speaking about models of dynamic functional connectivity. It's quite a complex and large field, which I don't intend to cover completely, but I do hope to give a few of the important concepts. As an introduction, I'll just step back and show this sped up video of a colleague of mine, watching a film actually, but I think it demonstrates that even at rest or doing daily tasks, humans are intrinsically dynamic and fluctuating. And it seems amazing that we've had to come along in the field for some years to reach the point where we're now really focusing on the nature and causes of these dynamics. In this course, I'll be speaking in very general terms about models of complex systems. I'll then walk briefly through how to make a model of a large-scale brain system, or a large-scale brain model, sorry. I'll give an example of something called metastable brainwaves, and I'll briefly introduce some of the simulation toolboxes for doing this yourselves. So an analogy, right? Why do we need a model of the brain? Why don't we just do complex and detailed measurements of the brain, as neuroscience has been doing and uncovering interesting things for many years? As an analogy, imagine that you wanted to go outside. You looked out the window and you get a picture of the outside world. You look up in the sky, and of course the sky is dynamic and you see these moving clouds. You're wondering though, in a couple of hours, is it going to be sunny and should I go to the beach, or is it going to be raining and should I take an umbrella? So you want some weather forecasting. Looking out the window isn't going to be sufficient. And how do you do that? Well, us citizens can just go and look at the Weather Channel or the Bureau of Meteorology. This is as sophisticated as weather forecasting gets now. This is a visualization of a global weather forecasting system called windy.com. And you can see in this instance, I'm cycling through wind, temperature, waves. So it's this, here's waves, a very beautiful multimodal picture of what is happening, not just outside the window, but everywhere across the region, or in fact, the entire world. So how does one of these beautiful forecasts work? Certainly much more than just a website. Well, meteorological models work by breaking the world's atmosphere into tiny little boxes or little grids. It's a bit like boxes of the brain, really. And in each of these, there's a local model of heat, temperature, and wind, or also in the ocean, ocean currents. So models of gases and their movements. And these small boxes are all simulating something called the Navier-Stokes equation. This is a model of fluid dynamics, which was basically derived from Newton's 3 laws, but customized to deal with incompressible fluids or slightly compressible fluids. So this complicated looking equation here describes the movement of fluid or gas or air, not gas particles, but the whole gas itself, through different volumes under different forcing terms, like heat, like the movement of adjacent boxes. So all of these little boxes are coupled together. They're then implemented on massive supercomputers, and they're run around the world. There's about 3 of these supercomputers. And these are used for planning flights, shipping. You can use it recreationally. I use it to predict when I should go surfing. Okay, it's one thing to forward predict, but you need to constantly update these models according to what's going on in the world. So simultaneously, there's these multi-grid weather recordings being obtained and constantly updating the accuracy of these models. This is something called data assimilation. Okay, so there's the model of Southeast Australia, and this is the framework in which it occurs. A model which forecasts and then is updated according to measurements. So there's a generative model, and if we can do it for the weather, can we do it for the brain? Can we have a generative model of the brain, and can we use it to forecast and understand brain states? So the answer, of course, or I wouldn't be here, is yes, we can. And I'm going to introduce some of these models. But the idea is that there is a similar type of equation, very different in its actual form, but a similar sort of goal, but not modeling gas and fluid, but population level firing rates and synaptic barrages and neuromodulation. And just as we can embody the weather in an equation, we can embody the flow of these different neural states in different equations. And these are generative models. And at the end of the day, they're very similar, whether they're describing the weather or the brain or a magnet. The form is different, so the terms are different, but the goal is very similar. With a generative model, that's okay for predicting what's happening in the brain or the weather, but you need to observe it and update it. So just as we do that for the weather, we have observation models for the brain. So we predict how neural activity maps into scalp EEG or into bold signal fluctuations. And actually, the science of developing observation models is just as important and just as challenging as the science of understanding the generative models. And likewise, ultimately, we would like to do brain forecasting. So we would like to have a really good model that forecast brain states and measure brain states, and that includes behavior, of course, and use these to update the model. And if at the end of this you think, oh, well, Michael Breakspear's models aren't very good, then you're very welcome to work with colleagues and develop an alternative model. And we can have a test to see which model works the best, the Breakspear or others models, or a new model. And I'll get a little bit onto that, but it's basically minimizing the error between the forward forecasting or the predictions and the continual stream of measurements. Okay, so now I'll just spend some time focusing on this big project in the brain. Now, since the 1950s, we've basically had pretty good mathematical models of single neurons. And these are called the Hodgkin-Huxley model, after two neurophysiologists, Hodgkin and Huxley. I don't want to go into the details of these equations, but they describe the flow of ions through ligand and voltage-gated channels in the cell. So these ions being sodium, potassium, leaky channels, maybe calcium. And this model, which was developed with careful observations of the single cell, actually the squid giant axon, works fabulously successfully for understanding and predicting and forecasting the behavior of individual neurons. And in fact, it earned Hodgkin and Huxley the Nobel Prize. But individual neurons, I shouldn't say neutrons, neurons, are buried within circuits of thousands of neurons, which are themselves immersed in functional subsystems and large-scale brain networks. So there's this hierarchy of scale, and we certainly wouldn't try and model the whole brain by modeling individual neurons. For a start, each neuron has 9 equations, and there's billions of neurons, so it's kind of unachievable. And also, human behavior doesn't, at the end of the day, depend on individual spikes, or we would be very unpredictable. We believe it derives from the collective action of many thousands of neurons interacting through axonal connections. Individual spikes are very stochastic, and their precise timing is far too noisy to code complex but reliable human behaviors. So it seems increasingly likely, and there's a good theoretical framework to believe it's the mass action or population coding that is important for perception, motor behavior, social intelligence, et cetera, et cetera. And also here at OHBM, we're looking at large-scale recordings, and these population models are very well matched to the apertures of fMRI, EEG, and MEG. So here's a local neural population, and if there's only hundreds or perhaps thousands of neurons, we can code up and simulate population activity. So here's a simulation of excitatory and inhibitory neurons, and this is their so-called spike raster, or the little dots record each action potential. And from these, if we want population activity, we just bin this population into little time bins, and then we look at the distribution of membrane potential. And that's one snapshot on the left and a later snapshot on the right. Now, the key thing is a reduction of the actual histogram of states to a simple probability distribution, and in this case, the normal probability distribution. This is called a mean field approximation. And so we're getting rid of all these states, and we're just going to model the mean and the variance, or the value of this population and the precision of that value, if you're thinking in terms of representations. Now, instead of having, in this case, 2,000 equations, we want a couple of equations. 3 or 4 maybe, that map these population distribution forward in time. And if it actually predicts the empirical distribution, then we have a good model. This is not an exotic or unusual approach. Physicists use this very widely in physics, this so-called mean field approximation, for all sort of collective behaviours. At times, we may not even be interested in the variance. We might just want to map the mean. Does the firing rate increase or decrease following a complex task in this population? This is called the neural mass approximation. Many others have derived this over several decades. You can read about it in a recent review paper that I wrote. What are the principles of derivation here? Is to take the complex, still barely understood neural tissue and power it back to some key cells in a small column. This might be a detailed multi-compartment model, such as the Human Brain Project is pursuing. We might even then simplify it even further. So each neuron is just like one little system with a couple of equations. Brian, neuron, and SpikeNet do these. Or we might just, as I spoke about a moment ago, get rid of all the individual states and just do a population model. And these are some of the well-known population models, Janssen-Ritt model and the Wilson-Cohen model. And I'll speak about some more. This is a mathematical mean field reduction, which is used very successfully in physics. We can also take a lot of the beautiful biochemistry of the individual cell, such as second messenger mediated neural modulation, sort of messenger systems from monoaminergic neurons that change the properties of the dendrite or the property of the cell. This is neuromodulation at the cellular scale. And we, just as we can express this in mathematical form for an individual neuron, we can do a sort of mean field approximation and we can get similar population responses. So we can look at the response of a population to an input, and we can look at how that varies as a function of different neuromodulators. And likewise, we can look at the gain or the output of a population of neurons. And Max Shine and others and I just recently published a review paper on that. Okay, so now let's build up our neural brain model. We have our evolution equation. We have a function that defines a vector field. This is a sort of segue into maths. At each possible point in this phase space, we get a collection of vectors. This is how the system will change under our evolution equation. We then get these orbits that connect up the vectors, and these describe flows and attractors. An attractor then is what the system will settle down onto after a few seconds, and the shape of the attractor will tell us about the type of behavior. So this is a limit cycle, which is giving an oscillation. We can have a more complex limit cycle that gives more complex oscillations like bursts and spikes, or we can have something called a chaotic attractor that will give us very complex arrhythmic activity. This is an example of a neural mass model, a rather old sort of toy model that we played around with for a while. Here are the time series on the left, and this is its attractor. Here it is in a bursting state. This is a population of pyramidal cells and inhibitory cells with slow potassium conductance. And so this is a bursting state. If we change the underlying parameters, then we get the same system with the same equations exhibiting this type of complex dynamics called chaos. Now we're going to pursue an approach like our weather forecasting colleagues. These are little patches of cortex or subcortical populations. We then connect them, but instead of interacting with their nearest neighbours, as with the weather, they're going to interact through long-range white matter connections. So here's two interacting. Here's a chaotic oscillator at one. Here's a whole bunch of these chaotic oscillators. Then using data from connectomics, structural connectivity, will inform how we let these individual populations interact. And then we can just fire it up on a supercomputer, or even a good desktop will do at this sort of coarse-grained approximation. And we can look at the forward evolution of this system. So many of you may have heard of work going on by quite a number of us in the field. Chris Honey, Olaf Sporns and I, Gustavo Deco and Victor Yersa, Peter Robinson, Petra Ritter, Leonardo Goyo, and many others have been working in this field. This is a model I did with James Roberts, exactly what I discussed a few minutes ago. We then got some connectomic data from healthy older Australians, and we asked ourselves, well, we don't really fiddle with the parameters. What kind of whole brain dynamics do we get? This is like the weather patterns I spoke about. Here we've got an example of large-scale rotating waves that emerge. Here is a few minutes later, the same system shows something we've called breathers. And this system can also show traveling waves. So these are collective behaviors that we observed from these population models. Waves have been observed in the laboratory across many different scales, invasive electrophysiological recordings, behavioral recordings, surface EEG. And so we were quite excited when we thought this. We thought, well, here's a model that others haven't really come up with before for spontaneous, highly nonlinear waves. And they're not magic, these waves. They come because the local connectivity drops off quite quickly, and it has a sort of what we call a spatial kernel. So if I'm active and I'm one of these populations, I'm more likely to change the activity in my neighbours and their neighbours. And this simple process gives rise to these large-scale travelling waves. We were able to do a qualitative comparison here to some surface recordings from Terry Sanowski's group. These are spindles, so waves of spindle activity acquired from people at sleep, and just showing the same sort of aperture, very similar carrier frequencies, very similar propagation velocities between what has been observed and what we modeled. So this is good, right? This is like coming up with a weather model that actually predicts the weather in advance, or the type of weather. Just want to close off all this about dynamics. That's one example. I won't have time to walk through others. What I showed was how these waves spontaneously went from the breathers, the traveling waves, the rotating waves. So the patterns are jumping between different types, and it could be a multi-stable system in which noise drives it from one attractor to another. It could be a metastable system, which sort of cycles through what's called ghost attractors. And you can also get criticality, which is when there's not really any attractor, there's just a lot of noise, and the system sort of wanders around like it's in a ghost state. And we've looked at these, and if you get into this field, I do recommend that you learn the difference between criticality, metastability, and multi-stability, because they're often used interchangeably, but they're really quite different. So to come back to the waves, we have some snapshots here of the states at 4 different times, using the Hilbert transform, we can then look at gradients in these states, and using streamline technology, as our colleagues in connectomics do, we can then look at the underlying dynamic flows. So these are these three different ways of looking at the same system: the waves on the left, the vectors in the middle, and the streamlines, the dynamic streamlines underneath. And what you see is quite a complex pattern on the right with these sinks and sources in different regions of the brain. And every now and then they collide, and we just saw the sudden emergence of a vortex. So this is what we believe gives rise to brain dynamics, and therefore gives rise to what you observe, namely dynamic functional connectivity. This will do me here. The two big simulation toolboxes for looking at population dynamics, highly recommend the virtual brain. Many of you may have heard of it. It's a very comprehensive Python open simulation platform for doing everything from epilepsy modeling, resting state, multi-scale. This is the brainchild of Randy McIntosh, Petra Ritter, and Victor Yersa. So I highly endorse the virtual brain. I've had some involvement in myself. If you're a MATLAB maven, and I know there's not many around these days, but if you'd like to learn more about this and you have access to MATLAB, our brain dynamics toolbox can be downloaded. And this is a simpler sort of interactive forum for understanding the sorts of things I've shown in the talk today. It can also scale up, and in fact, these whole brainwaves I just showed were generated in the Brain Dynamics toolbox. It gives you a nice user interface with these phase portraits, time portraits, and the equations. And it comes shipped with a whole lot of models, or you can go behind the toolbox and watch some educational videos that Stuart Heitman has put online, and you can code up your own models. Once you've done that, running the model is exceptionally easy. It's basically 2 lines in the command window, and you can play with the parameters, and you can get a really nice understanding of what type of dynamics any particular model will generate. It's open source, so you can download it for free, add it to your path, and in 10 minutes, off you go. I'd like to acknowledge funding for the work of my group, the ARC, through the Centre of Excellence in Integrative Brain Function, the NHMRC. You can download the Brain Dynamics Toolbox. Actually, I think it might have moved from there. And we've got data and simulations available as well.
Hello, everyone. It's a pleasure to be part of this Dynamics Educational Symposium. For my presentation today, I'd like to tell you a little bit about our work looking at timescales of variation in functional brain networks across multiple different time spans. Now, our ability to map functional brain networks with fMRI has greatly improved in recent years, as I'm sure many of you are aware. And with sufficient data, we now know we can map these networks very reliably, even at the level of individual participants. Excuse me. Now, at the beginning of the talk today, I'm just going to go over some of the basic methods to make sure that we're on the same page about the terms that I'll be talking about today. So when I talk about functional connectivity, what I mean are correlations in the bold activity over time between different brain regions. So for example, if I measure the bold activity in left and right motor cortex over time, I'd see that these regions are very highly correlated with one another. A region that's part of a different system, like left visual cortex, may show much lower correlation patterns. Now, what's somewhat surprising when this was first observed is that these high correlations are seen not just when we ask participants to complete a motor task, but also when participants are sitting as still as possible at rest, yet the spontaneous activity patterns in left and right motor cortex show very high correlations. And these spontaneous correlation patterns mimic the patterns of evoked activations that we see during a motor task. Now, these principles are true not just for the motor-related regions I showed you on the previous slide, but many different regions spread throughout the brain. I can show them to you all at once in what's called a correlation matrix, where any given cell in this matrix just represents the correlation between a pair of brain regions. Now, if I order the regions randomly, it's hard to see much of a pattern in this matrix. But if I reorder them such that I place regions that are part of the same system close to one another, you see this really beautiful structure emerge, which is called the block diagonal structure, where you have high correlations along the diagonal and low correlations in the off diagonal. Now, because of this structure, we can then quite effectively use different community detection algorithms to identify the systems of the human brain, which I can mark off with red lines as shown here, and paint on the brain in different colors as shown here. Now, this is very exciting because we've now used a data-driven technique based on spontaneous brain activity to map systems of the human brain. And while this may seem relatively unconstrained, a number of studies have shown that this can produce very robust patterns of the typical network architecture of humans, as shown here for two large group studies, one at WashU on the left and one from Harvard on the right. And even though these were collected on different participants, different scanners, and using different community detection techniques, you can see that they produce very similar pictures of what the typical networks are of the human brain. So this has led to a lot of excitement in the field for our ability to use functional connectivity to address different types of questions. For example, how functional systems are affected by brain damage and disease, how they may differ across individuals and linked to individual traits, how they may change over the course of the lifespan with, say, development and aging, and how they may vary across different cognitive contexts or with different cognitive demands. However, these different types of questions differ in their underlying assumptions about the relative stability of functional networks. For example, some of these applications suggest that we would think functional networks are primarily stationary, so we can make a single measurement, and that measurement can tell us important things about that person's disease status or trait-like characteristics at other moments in time. Or they suggest to us that functional networks are constantly reconfiguring and reflecting ongoing cognition, mood, arousal, and so forth. So as it may sound, these qualities can have important impacts, both on our interpretation of differences that we see between groups or individuals or conditions, suggesting whether or not they're more likely to be driven by ongoing cognitive state or more stable factors. And also for our ability to use these functional networks in various different settings. For example, in personalized medicine settings where we might want biomarkers that show relative stability over time and not be affected by, say, the mannerisms of a technician or whether someone ate breakfast or not. And although I present these two hypotheses as being two extremes, there's actually a spectrum on which we think that functional networks might vary. On one end, you might think functional networks are completely invariant and that the only differences in these network patterns that we observe are due to sampling variability. And on the other extreme, you might think that they should vary constantly from moment to moment as we have different thoughts, as our neurons are firing at different rates, and so forth. We could also think that functional networks will change more slowly over the course of minutes as we enter into different brain states, such as task states or states of arousal. That they may change more slowly over hours or days, such as with circadian rhythms. That they may only change over weeks or years, either with natural developmental processes or extensive experience in a particular task. Or that they may be fundamentally stable within an individual, but vary meaningfully across individuals. So my talk today, I'm going to review these different concepts. In the first part, I'm going to focus on past evidence for and against stability and functional network measures with a cautionary note about potential other causes of apparent dynamics in functional connectivity. In the second half of my talk, I'm going to use a precision fMRI dataset to contrast the relative magnitude of functional network changes across different timescales, with precision fMRI referring to fMRI where we collect many different measurements from a single participant over time. Now, before I get into this data, I want to make a couple qualifications about what I'll be talking about today. So all of these data sets are based on functional MRI, which of course is slow and may be different from other kinds of measures like EEG or single unit recordings, which capture very different timescales of neural processing and may have different statistical properties. Also, when I talk about dynamic changes in functional connectivity, what I'm talking about is variation in the correlation patterns of different regions. That is the second order statistic, which is different from talking about variations in the first order activity patterns themselves, which are related, for example, to evoked responses during a task. So first, let's go over and tackle whether or not there's evidence that functional connectivity changes across different states, such as periods when you're engaged in a particular task or changes in arousal. So when a number of groups, including ourselves, have contrasted functional connectivity between task and rest, it has consistently been found that functional connectivity can vary systematically. For example, in this study, we looked at how functional connectivity changed across 3 very different cognitive domains relative to rest. We see that there are differences both within network and functional connectivity patterns. You can see the relative increases in default mode functional connectivity and decreases in visual network functional connectivity, as well as changes in between network functional connectivity patterns. Moreover, in the study, we presented evidence that these changes were separately related both to the evoked activation properties of different regions, as well as their network hub-like characteristics. We also showed that particular tasks can vary meaningfully from one another, as shown here for the 3 different tasks, which is also supported by the fact that machine learning studies can be used to have shown that you can very accurately decode task states from functional connectivity patterns. This can also be seen with variations in states for much simpler tasks. For example, just opening or closing the eyes can cause differences in functional connectivity. So we see that there is evidence that functional connectivity can change across different task states. What about during rest, where at least subjectively, many of us have the experience that we're thinking about different things while we lie inside the scanner? So this is a study that I'll be telling you about that was led by Tim Blauman. And this is perhaps most commonly been studied using sliding time window techniques combined with some form of clustering, such as k-means clustering, to examine if functional connectivity changes over different time windows and how they might be related to changes in underlying cognition. So this data suggests that there may be additional structure to functional connectivity at rest that may accompany our subjective feeling of changing thoughts during this time window. However, I would like to caution that care needs to be exerted in the measurement and interpretation of dynamic functional connectivity, because unlike task modulations, there's no external imposed perturbation, which means that it may be hard to distinguish intrinsic variability from other sources of variation that might be noise in this context. For example, sampling variability, motion and respiration, and perhaps arousal, and whether or not that's noise may depend on your specific question. I'll go over each of these in the next few slides. So by sampling variability, I'm just referring to the variability that you probably learned about in one of your first statistics classes. That is that your sample is just an estimate of a statistic from the full population, and that this sample estimate will have a certain variability or error associated with it. The error goes down with increasing samples from the population. And the same has been shown to be true of functional connectivity measured over time, which shows a clear progression of decreasing error with additional time points. Since dynamic functional connectivity is based on a small amount of data, we need to be particularly careful about this type of error. So how can you identify patterns that appear dynamic but are just due to sampling variability? Well, one way to do this is to construct an appropriate null model that is fully stationary but just includes random noise, and monitor whether it also shows putative dynamic patterns that are similar to what you see in your real data. And so Tim in his study did this by creating a simulation where he started with just a random noise time course, and then imposed on this time course a static functional connectivity pattern similar to what we see in real data as shown here. And also enforced that this functional connectivity time course had a similar power spectrum to what we see in true data as shown here. He then compared the simulated null model to what we see in true real bold data. So here I'm just showing you 2 movies, one from the simulated data and one from the real data. And I would argue that if you look at this over time, it may be hard to distinguish between the 2. And in fact, now I can put them on and you can see that the one on the left is from the simulated data, the one on the right is from the real data. But you can see both show substantial fluctuations. So with this model, Tim used the same sliding time window technique and k-means clustering approach to identify different states in both the simulated, that is the stationary data with noise, and the real data. And if you look here, you can see that also in the simulated data, it's also possible to identify different states. And in fact, these states can look a lot like what we see in real data. And this suggests that the presence of states alone can't be taken as evidence of different state changes in dynamic functional connectivity because they can be driven by noise or sampling variability. If you're interested in comparing this model to your own data, I'm putting up a link here with the code to generate it, and do feel free to reach out if you have any questions. What about other more systematic biases in functional connectivity? One way to think about, one that might immediately come to mind is motion, as its transient nature may make it look a lot like a dynamic process. And when we look at motion more closely, we can see that motion does indeed correlate with the kurtosis of functional connectivity, where kurtosis is the variance of this covariance measure, the fourth moment. And so removal of motion through censoring or scrubbing techniques can remove most of the variation that we see in functional connectivity, as shown here. A second thing that we might look at is arousal as we get more or less awake inside the scanner. And what we can see is that the remaining portion of the variance may be explained by arousal, indicating that those entering into lighter sleeps have heavier levels or higher levels of kurtosis than those that are fully awake. Also note that Tim didn't look directly at respiration-related effects as shown here, but respiration, especially related to deep breaths, may also induce transient patterns in functional connectivity that may inflate dynamic statistics. So here I've shown you evidence that functional connectivity can show significant modulations between task states, but also that variations in functional connectivity within a single resting state can be easily confounded with other factors, such as sampling variability, motion, and arousal, making it difficult to find resting state variation that might be linked to cognition. Moreover, there's also evidence that introduced at the start of the talk that functional connectivity can show some degree of invariance across large groups. So can we put these effects together in the same sample to ask how they compare and what their relative magnitude is, and thereby speak to the relative value of functional connectivity to address these different types of applications I introduced earlier? So this brings me to the second part of my talk. So for this data part of the talk, I'm going to be talking about a precision fMRI dataset called the Midnight Scan Club dataset, which was spearheaded by Nico Dosenbach, Steve Nelson, and collaborators. Now, this dataset is really well suited to addressing this question because it contains a lot of data from 10 different participants. So each of these 10 participants completed 10 different functional MRI sessions each. And with this large amount of data, we can now show that we can map networks very reliably at the level of these individual participants, shown here on the outside, relative to the group average shown in the center. Now, in this data, from each day, participants completed not only a resting state scan, which was used to map the networks as shown in the previous slide, but also 4 different task scans. A semantic task where participants were asked to complete a noun-verb judgment, a coherence task where participants were asked to say whether or not dots were arranged concentrically on the screen, a motor task where participants were asked to move their hand, foot, or tongue, and an incidental encoding task where participants were asked to categorize faces, scenes, and words that were repeated over time. Now, this design is really nicely suited to looking at the system, how functional networks may vary, and in particular asking whether we can separate effects that are either common to this full group of data sets or selective to particular subjects, to particular days, or particular task states. So we measured functional connectivity matrices from each particular participant, shown here in the 2 rows, from a given session and task, as shown here, using a background connectivity approach. And we looked at the similarity of these data sets using a few different approaches. First, I'm going to show you a data-driven approach where we use multidimensional scaling to plot the similarity between these functional connectivity matrices. Now, in multidimensional scaling, each of these dots represents one of those correlation matrices I showed you on the previous screen, and their position is dictated based on their similarity. So the closer together that they are, the more similar they are to one another. Now, the two graphs I'm showing you here on the left and the right are exactly the same. I've just colored the dots differently, such that the ones on the left are colored based on subject identity, and the ones on the right are colored by task. And so what you can see here is that most of the variance in the data is organized by subject identity rather than task state at these first two dimensions, which explain most of the variance in the data. And only at higher dimensions do we start to see divisions based on task. So you can see the motor task clustering around dimensions 8 to 9, and then the other 4 tasks clustering separately from one another at dimensions 10 and 11. We can also quantify these effects a little more directly by using what's called a similarity matrix, as shown here. Now, this is a second order matrix where any cell in this matrix just represents the similarity between a pair of functional connectomes, again, from a particular subject, task, and session. I've organized these with subject identity as the primary organizer along the diagonal, within that by task, and then within that by session. So first, before we look at the actual data, I'll just use these cartoons to orient you to what we might expect to see in these patterns. So effects that are shared across this full group of data sets would increase similarity across this full similarity matrix. Effects that are specific to a given individual will be seen along this main block along the diagonal. And effects that are specific to particular tasks will be seen in these little mini blocks along the diagonal and the off diagonals relative to that. And of course, you might get interactions between different patterns. For example, effects that are specific to an individual and task in the single mini block along the diagonal, or a particular individual and session, which will be seen in this checkerboard pattern. So if we return to the actual data, what we end up finding is a mixture of these different effects. So first off, you might notice that the background is not centered around 0, but rather around 0.5 or 0.6 correlation, indicating that there is a substantial amount of similarity shared across all of these functional connectomes. Now, relative to this common pattern, we also find a really big effect of individual. You can see in this main block along the diagonal, indicating that a particular person's functional networks look most like themselves, regardless of the day or the task that they're completing. There is a common effect of tasks. So say everyone doing the resting state looks a little bit more like others doing during resting state, but this effect is small and hard to see on this color scale. Now, relative to the individual pattern, you might notice a little bit of a pattern along the main block of the diagonal. This indicates that there is an interaction between individual and tasks, suggesting that a particular person doing, say, the motor task will look most like themselves doing the motor task rather than another task. There is also a small effect of day-to-day variability, but this can be hard to see on this color scale. Now, I want to make 2 points here. First, much like past literature has suggested, all of these effects are present in functional connectivity and highly significant. But if you contrast them directly, what you'll see is that they differ substantially in their magnitude, such that functional connectivity matrices are really most driven by stable factors, such as commonalities across the full group, as well as individual specific features, with day-to-day or cognitive variability only showing much smaller effects, interestingly, many of which interact with individual variability. We also looked at how these different factors varied across the brain. One way we looked at this is by contrasting higher level association networks involved in top-down control, such as the frontal parietal cingulopercular networks, as well as the dorsal and ventral attention networks. And we contrasted these with more basic sensory motor processing systems like the visual system, the auditory system, and the somatomotor systems. And when we look at the patterns from these two systems, we show that they have a similar overall pattern, but do differ significantly, with individual specific effects that are shown over here on the right side of the plot being larger in control systems than sensory motor processing systems. And the opposite effect, cross-subject effect, shown over here on the left side of the plot, being larger in sensory motor processing systems than control systems. And so this is consistent with past results, suggesting that these high-level control-related systems are those most affected by individual variability. We also looked at whether or not these effects were consistent across a number of other spatial scales. For example, contrasting the functional network effects for individual parcel brain regions that we looked at, as well as for individual connections. We saw similar patterns at each of these scales, where we see that the functional connectivity pattern is dominated again by stable factors, either group or individual features, with only more minor contributions from task and day-to-day variability. I also want to return to the qualification that I made at the start of the talk, where we contrasted that most of what I'll be talking about today are variations in functional connectivity rather than variations in the first order activation response. And so I want to note that, again, when we make these similarity matrices for these two types of measures separately, we see that these properties are really driven by functional network properties that show very high stability over time, whereas evoked signals show much more variability and over time, and in particular sensitivity to task state, as may not be surprising given the wealth of literature using evoked responses to understand cognition. Moreover, while at the beginning, I also pointed out that the properties of these studies that I'm telling you about today are specific to fMRI, which is slow. An intriguing recent paper from Kurhan Mostame and Sepide Sadagiani's lab suggests that this high level of stability in functional connectivity or functional architecture across states may also be true of phase and amplitude coupling connectivity estimates in ECOG data at many higher frequencies, as exemplified in this figure below. So let me just summarize what I've shown you here. I've shown you some evidence that functional connectivity patterns are really dominated by stable factors, either shared across large groups or specific to individuals. There is also variation in functional networks from day to day, as well as across task states that can be highly significant, but these effects tend to be much smaller in magnitude. And finally, these effects differ somewhat in their anatomical distribution, with group level patterns being strongest in sensory motor processing systems, individual specific features having the biggest effect on top-down control systems, and task effects, and especially individual by task effects, showing a mixture of these two different patterns. Now, I want to close this part of the talk by leaving you with some thoughts about why I think these findings are important. First, because I think they suggest something about the potential neurobiological contributions to functional networks, suggesting that much of what the signal that we measure in functional connectivity is driven by very slow or stable processes, rather than what we're currently thinking about at a given moment in time. This, in turn, tells us how we might interpret functional network comparisons between groups and states, suggesting that when we say measure a difference between younger and older adults, or a disease group and a non-disease group, that it's most likely driven by stable features that are different between these two groups, rather than their ongoing cognition in the scanner. And that indeed, if we're interested in how state-like differences between these two groups, that we might want to carefully account for these stable features. One of my graduate students, Alexis Porter, is currently investigating this question using machine learning to decode task state and showing that when we create our machine learning patterns to account for individual specific features, we get a big boost in the accuracy relative to machines that are forced to classify across different individuals, suggesting there's a lot of individual specific information that one can take advantage of in this case. Finally, I think these results are exciting for the potential for functional networks in different medical applications, for example, in the development of biomarkers or in neurosurgical planning. Because of their stability and their sensitivity to individual differences, it suggests they're really well suited for these different techniques. This is something we're actively pursuing in the lab. I just put up that one of the domains that we've been looking at this is in Parkinson's disease, to try to understand the widespread network effects that occur in this disorder. So I just want to summarize here. In the first part of the talk, I showed you some evidence for and against stability in functional networks, showing that there is significant change in functional connectivity across different task states, but that caution needs to be taken in interpreting functional connectivity's changes across rest, given other sources of potential dynamics. And in the second part of the talk, I contrasted the relative magnitude of variations across different timescales showing you evidence that functional connectivity can be quite stable over time, and that we might want to carefully account for stable factors if we're interested in more transient dynamics. So I want to end this talk by noting a few conclusions that may be relevant. So first, as I'm sure you've been hearing across all of the talks in this session, I think it's important when we talk about dynamic functional connectivity to define the terminology and the constructs that we're thinking about clearly. Secondly, if we're looking at fast timescales of variation in functional connectivity, it may be important to address other potential sources of variation. This can include variation in sampling variability. I showed you one way to take care of this is with a null model. Raphael has some very nice other null models that can also be used in this domain that he may have talked about earlier. Another thing to note is that you may want to carefully account for physiological forms of variability, such as from motion or respiration through appropriate denoising. Here I'm just putting up some papers which talk about different denoising techniques and contrast their ability to remove these different types of signals. And lastly, we may also want to consider whether or not any patterns of variation that are remaining in our data may be related to variation in, say, arousal rather than ongoing cognition. This may be of interest to you, but it may also not be of interest to you. And so I think either you may want to either account for it or incorporate it into your interpretation of different results. And finally, I'd like to make a plug for collecting substantial amounts of data for individuals moving towards this more precision fMRI technique in order to best assess both individual properties and variation in the individual properties of functional networks over time. And so I'm going to put up here just a couple links for those of you who are getting started right now with functional connectivity of 2 different tutorials based on precision fMRI, one in MATLAB and one in Python, that you might want to look up if you're interested in learning more. Because this is an educational symposium, I want to leave you with a few reflection questions that may be relevant to consider when embarking on a project on variation in functional connectivity. And so the first is, when you're looking at variation in functional connectivity, you may want to consider how variation in functional connectivity, is related to ongoing cognitive content versus arousal or other neurobiological phenomena, and how this may relate to your particular question of interest. Related to this, second question is to always carefully consider what uninteresting causes what might be uninteresting causes of dynamics in your data, and to carefully account for these in your analysis procedures. Many people have come up with clever ways of addressing these. And the last one is a broader context of trying to understand how you can demonstrate both the reliability and the validity of dynamic functional connectivity, which is a tricky question because it inherently varies over time. And so one question is, what is the appropriate null model for your particular question on dynamic functional connectivity? And this will depend on what sort of assumptions and conclusions you're trying to make. And so I want to close by thanking my collaborators on the different projects that I talked about today. And in particular, I want to mention Tim Lauman and Steve Peterson, who led a lot of the initial dynamics work during REST that I mentioned in the first half of the talk. I'm also going to put up my contact information here in case you want to reach out to me individually, and I'll be available at a question and answer session afterwards. Thanks very much.